Okay, I think so we can start, but I can't see Dr. Adina yet. So maybe I already sent her an email. Yeah, so in case if she's late, maybe Dr. Chen can present first. Hello, Professor Chen, can you hear me?
Yes, sorry. Yeah.
Okay. Yeah.
Sorry,
so
should we start now?
Yes, we can start now. If Dr. Adina is late, you can present first. I mean, as the second speaker.
Okay. Okay, no problem.
Yeah. Okay. So then, yeah, I'll start first, and then I'll hand over to you. Okay. So good morning, everyone. Welcome to the Symposium Microbiomics from Bench to Chairside. We have three excellent expert researchers in the field today. We have Professor Kimberly Klein, who's an Associate Dean for Faculty Affairs in the College of Science, Professor of Microbiology in the School of Biological Sciences, and a Principal Investigator at the Singapore Center for Environmental Life Sciences Engineering. She has received multiple awards for her contributions to the field of microbiology, including a NIH K99 Career Development Award in 2011, the Singapore National Research Foundation Fellowship in 2011, the ICAAC Young Investigator Award from the American Society of Microbiology in 2014, and the Nanyang Education Award in 2017. And our second speaker is Dr. Noor Adina, an associate professor in the field of periodontics in the Faculty of Dentistry, University of Malaya, Malaysia. She has 17 years of experience in research, teaching, and management roles. She has been a principal investigator in various research projects pertaining to obesity and periodontal disease, periodontal systemic relationship, regeneration, and animal models. Our third speaker is Dr. Evan Chen, an associate professor of Graduate Institute of Clinical Dentistry, National Taiwan University, and the attending physician, Department of Dentistry, National Taiwan University Hospital. She has conducted numerous clinical and translational research projects, particularly on the microbiological and immunological aspects of the associations between periodontitis and systemic diseases. Recently, Dr. Chen has identified the specific oral microbiota in patients with Alzheimer's disease with next-generation sequencing. Dr. Chen has been awarded the first prize in research competition at the 2017 ITI World Symposium, and she has been nominated as ITI Fellow, Director of ITI NTUH Study Club, and Chair of ITI NTUH ITI Scholar Center. Dr. Chen is also the chairperson of the symposium today. The stage is yours, Dr. Chen.
Okay, thank you very much. Good morning, ladies and gentlemen. I'm Ewan Chen from National Taiwan University. So today I will be the chairperson. The symposium title is Microbiomics from Bench to Chairside. So at first, I would like to introduce Professor Kimberly Klein. Her research work is to understand the bacterial virulence factor which contribute to colonization, bioinformation, and the infection of E. faecalis and the related pathogens. So let's welcome Professor Kimberly Klein. Thank you very much.
Morning, thank you. Can you see my slides?
Would you please share your slide? I couldn't see your slide.
I am sharing them. Can you see them?
Yes.
No, it's okay.
Okay, great. Okay, great. So thank you again for the introduction here. And greetings from Singapore. Today I'll be telling you, as you just heard, about polymicrobial infections. And so we approach this, and I should say this is a really microbiological talk, and I realize the audience here is broad, but what I hope you can take away from this is some lessons about how to think about mixed bacterial species interactions in the niche that you're most interested in, which I think is the oral cavity for most of you. So we think about biofilm-associated infections in a much broader sense, and that's because most chronic infections are thought to be biofilm associated. And what that means is that when bacteria are growing in these dense bacterial communities that are biofilms, that confers antibiotic tolerance to the microbes within them. And moreover, many at least, biofilm associated infections are polymicrobial in nature. And that polymicrobial nature can further amplify antibiotic tolerance. So it can make these infections quite difficult to treat. So that's really our motivation. And so we take as our model organism Enterococcus faecalis. This is a commensal of the GI tract of probably everybody that's listening here, but it's also an opportunistic pathogen of a variety of sites in the human body. For your purposes, of course, the oral cavity is a major site of infection, as well as other places like the heart where it causes endocarditis. It's most frequently associated with catheter-associated urinary tract infections, and it's a frequent cause of wound infections. And so in my lab, ironically, for this audience, we actually model all of these infections except that in the oral cavity to study biofilm-associated infections caused by enterococci. In addition to that, most of these niches where E. faecalis is found in the human body are polymicrobial in nature, and that means there's other bacteria around during the infection. And so our real big picture goal is to try to understand how this bacterium senses these really complex host sites, both bodily sites and the microbial community there, to respond accordingly to build the biofilms that we know are essential parts of all of these infection cycles. And so today I'm just going to use the wound infection model as an example of how we can think about these infections. But all of the principles would apply to studies in the oral cavity as well. Okay, so how do we study these biofilm-associated infections? The traditional approach from a microbiological standpoint is to take a single species, your favorite bacterial pathogen of the oral cavity, for instance, grow biofilms on a plastic plate, and look for genetic mutants that don't make biofilms. And this starts to tell you how the organism makes biofilms. Then typically we can transfer those mutants to an animal model of infection and ask whether it's recapitulated in vivo. And what happens more often than not is that doesn't recapitulate. We don't see good translation from in vitro biofilms to in vivo. And we think that's because there's a lot of complexity missing in that in vivo or in vitro model. And for E. faecalis, this has been established in the literature using these in vitro assays to study biofilms does not necessarily translate to in vivo. So we have flipped this approach on its head. We start with complexity. We think about the polymicrobial community that E. faecalis is in during infection. We use animal models to look for human infection-relevant phenotypes in the animal, in this mixed species environment. And once we find that, then we move into our in vitro model systems to work out the molecular mechanisms of interaction, and we just use the animal models to validate what we've studied in vivo. Okay, so I want to just tell you a little bit about what we have done in the lab. So we always take it as our starting point, the clinical metadata from humans. And so here I'm showing you data from UTI and diabetic wound infections, but I could put up a similar pie chart for the oral cavity. We look for the organisms that E. faecalis is most frequently associated with during infection. And so you can see in both the case of wound infection and UTI, we see E. faecalis together with E. coli, with Staph aureus, in both sites. And so we then use that to try to make a more simplified system to study the organisms that interact in these microbial communities during infection. And so in the case of E. faecalis and E. coli, we've shown that E. faecalis can drive E. coli virulence, and we've worked out how that happens. We've also found that Staph aureus can drive E. faecalis virulence, and we've worked out how that happens. And in a very serendipitous observation, we found that E. coli can kill Staph aureus, and we've worked out how that happens. And I'll give you just a glimpse of each of these today in this talk. So in some published work, I'm going to summarize in one slide. Again, we've shown that E. coli virulence in this wound infection model is certainly heightened by the co-infection with E. faecalis. We recover higher bacterial burden, and there's more disease in the animal. We can model this with several in vitro biofilm models, and then use that to work out the mechanism. And what we find is something like this. We know that Ephicalis constitutively secretes the small molecule ornithine, which serves as a cue for E. coli to turn on iron acquisition machinery. And we're modeling all of this in a low iron environment, which is what the bacteria would encounter in the human host, particularly in infection conditions. So then Ephicalis is essentially telling E. coli or helping E. coli acquire iron under conditions where otherwise its growth would be restricted. So that's one example of a type of community that we can interrogate. And so you can look at that online if you're interested, since it's published. I want to say a little bit more about a second example, and that's looking at Ephicalis and Staph aureus interactions. And so here again, the take-home point is that Staph aureus can promote Ephicalis growth and biofilm formation. And this is work been carried out by a postdoc in the lab, Junhong Chung, who now is a faculty member at the National University of Singapore and continues this work there. And so what we found in this study is that, again, starting with an in vivo mouse model of biofilm infection, we find that compared to an Ephicalis single species infection, if we co-infect with Staph aureus, we recover significantly more Ephicalis from the infection site. Staph aureus numbers are not affected here, but so this is an Ephicalis specific benefit. We can again use in vitro biofilm models to study what's happening and mimic what's happening in the mouse model. And what we can, an example of this is that we can again recapitulate the increased Ephicalis numbers in the presence of Staph aureus in these in vitro models compared to Ephicalis monospecies biofilms. So again, we have a nice in vivo phenotype and in vitro phenotype that mimics that. And so then we can use this in vitro model to interrogate what's happening. Here we're looking at biofilm formation in a more classic assay. This was a crystal violet-based plastic dish assay, where again we see Ephicalis forms biofilms all right by itself. Staph aureus in these conditions doesn't make biofilms at all, but if we grow mixed species biofilms, we see this really heightened biofilm formation in combination. This is more than what would occur additively, so this is a synergistic effect. And what's important about this is this only happens within biofilms, within these densely packed bacterial communities. If we grow the bacteria under planktonic culture conditions, there is no longer a benefit to Ephicalis from Staph aureus. So we are bacterial geneticists, and we always take genetic approaches to try to dissect these really interesting interactions. And so when we did that, we looked for Ephicalis mutants that were no longer susceptible to this augmentation by Staph aureus. And we found mutants in a couple of genes that are important for aerobic respiration in Enterococcus faecalis. And so that's diagrammed here. And so how this works is that this MEN-A gene that seems to be important for this augmentation is important for the production of an electron carrier in the membrane of the bacteria that then delivers this electron to this terminal oxidase called SID-AB. And then this enables then the reduction of oxygen to water and the production of ATP. And one important thing about this enzyme SID-AB is that it requires exogenous heme from the environment to be active. That's these little red squares. Without that, without heme in the environment, this system cannot work. It can't go. So we, based on this finding, then we hypothesized that in this mixed species biofilm, respiration and Ephicalis aerobic respiration is being activated to drive ATP production to promote its growth in the presence of staph. And so then the prediction is that these augmented biofilms should be dependent both on oxygen, which is required for aerobic respiration, and exogenous heme. And so that's actually true because we can grow these biofilms, these mixed species biofilms under anaerobic conditions, and we no longer see the Staph aureus mediated augmentation of Ephicalis biofilms that we see under aerobic conditions. And moreover, if we take away the Staph aureus and just instead add sources of exogenous heme, we see that both hemin and hemoglobin can drive Ephicalis biofilm formation only under aerobic conditions, but not under anaerobic conditions. So then this led to the question of where is this heme coming from to activate this aerobic respiration enzymatic machine. And so we hypothesized that Staph aureus was donating the heme for Ephicalis. Staph can make its own heme, and this has been well characterized. So we could give bacterial mutants from staph that don't make heme and ask if they were still able to promote Ephicalis biofilms. And the answer to that is no. And really the important parts of this graph are shown here. Again, Ephecalis biofilms are really heightened in the presence of wild-type Staph aureus, but they are not increased in the presence of staph that can't make heme. So then we're left with this question of how does Ephecalis get the heme from staph to activate this aerobic respiratory chain? And so using some comparative genomics approaches for looking at Ephecalis strains that were not susceptible to this staph-mediated growth, as well as some really old literature suggesting that a secreted protease from Ephecalis could break down heme or hemoglobin, we hypothesized that a secreted protease called gelatinase, which many people that study Ephecalis biofilms are really interested in, we predicted that gelatinase might be involved in this heme degradation. And so here on the left is all of the data I've already shown you. For wild-type Ephecalis, it's augmented by staph, by hemin, and by hemoglobin in terms of biofilm formation. But a gelatinase mutant is no longer augmented by Staph aureus, is not augmented by hemoglobin, and is still augmented by hemin. And this makes sense because of these three sources, hemin would be the only one that doesn't need proteolytic degradation to release the heme that is bound by the proteins. So then altogether, this allows us to put together a model of how we think these organisms are synergistically interacting within a biofilm in a mammalian host. And that is that Staph aureus is releasing readily hemoproteins into the environment. And that gelatinase, which is only produced by Ephecalis in biofilms, which we think is why this is a biofilm-specific phenotype, this gelatinase then can break down these hemoproteins, release heme, which can be taken up by Ephecalis to activate aerobic respiration, which is more productive in terms of ATP production, and this allows its growth. So it's this heme cross-feeding from staph to promote Ephecalis biofilms. Okay, so that's the first example I want to show you. Now, in the second example, I'm going to move away from Ephecalis. So I'm going to get a little bit farther from the oral cavity. But again, the principles here, I think, could be translated. And again, this was a really serendipitous observation. And we found this when we were doing some controls for our Ephecalis work. And what JJ, a PhD student in the lab, discovered was that Ephecalis could kill Staph aureus. And again, these sorts of interactions are interesting because they then allow us to think about vulnerabilities and pathogens, potential avenues for new antimicrobials against important pathogens. So we always follow up these interesting observations. So again, this started with an in vivo phenotype, again, using our wound infection model, but you could imagine doing similar things with our other infection sites as well. And so compared to a Staph aureus monospecies infection, where we get very high, pretty severe infections in these wounds, if we co-infect with E. coli, and this dotted line here is the inoculum, the number of bacteria we deposit onto the wounds, you can see that Staph aureus is dying. It's not growing, it's not staying the same, it's actually dropping in the presence of E. coli. And E. coli numbers are unchanged in the presence of Staph aureus. So again, we can use biofilm models in the laboratory. These are macrocolonies grown on agar plates that can recapitulate some of the things that we see in the mammalian host. And so that's shown here. If we grow these biofilm macrocolonies, either Staph aureus alone or with the mixed species shown in the checkered boxes here, you can see that and the top dashed line is the starting number. You can see by 8 hours, the staph isn't growing in the presence of E. coli, but by 24 hours, it's all dead. We cannot recover any viable Staph aureus. So this is a really remarkable phenotype. So again, we are bacterial geneticists. So the approach that we take is to look for, here we were looking for E. coli mutants that could no longer kill Staph aureus. And what we found was that the vast majority of the mutants we identified mapped to this very large 54 kilobase pathogenicity genetic island called PKS. And so this is actually really well studied. It's found in many enteric bacteria. It is involved in the production of this polyketide non-ribosomally synthesized peptide called colibactin. Colibactin is particularly well studied and interesting to many researchers because it's highly correlated or associated with human colorectal cancer. So we know a fair amount about how colibactin acts on mammalian cells in terms of cancer, but it's really not been studied in the context of interbacterial interactions. Importantly, colobactin is a pretty elusive molecule. It's not very stable at high abundances, so it's been very difficult to study in the laboratory. So we don't have a lot of data about it. The other genes that we identified in this search for E. coli mutants that could not kill Staph aureus included this two-component signal transduction system called UVRY and BAR-A, and I'll come back to that in just. Okay, so once we identified these mutants, we wanted to verify that these were really involved in killing Staph aureus. And so here we're looking in our in vitro biofilm model compared to single species Staph aureus biofilms. If we add in E. coli, Staph is killed dead. If we add in a mutant of that very large PKS island, we see that this restores growth of Staph aureus, not to full single species wild type levels, but this is as much a restoration as one can get in a mixed species biofilm where the bacteria are competing for nutrients with the other infecting organisms. So this really is full restoration for all effects, for all intents and purposes. In addition, that BAR-A UVRY two-component system, this is involved in the killing of Staph aureus, because if we mutate those, we see a partial recovery of killing, not a full recovery, but a partial. So it says that these genes are involved somehow in the killing of Staph aureus. And we, like I said at the beginning of the talk, we can use what we discover in vitro and validate it in our infection models. And so here we're going back to that wound infection model and validating our in vitro findings, and they do hold up. So here again, Staph aureus is killed in the presence of wild type E. coli, but if we co-infect instead with that PKS mutant that doesn't make colobactin, Staph aureus wound infections remain at very high titers. And if we co-infect with mutants of the BAR-A or UVRY gene, we see a slight reduction compared to a monospecies infection, but certainly a significant recovery, again, indicating that this is involved in vivo during infection. So let me tell you a little bit more about colobactin. So this, again, has been worked out mostly with an aim for people's interest in human colorectal cancer. So it's synthesized non-ribosomally from the genes encoded at this PKS island into a number of metabolic intermediates, but the main product is this pre-colobactin molecule, which gets exported across the inner membrane of E. coli into the periplasm, where it's then cleaved into a precursor protein that goes away, and the mature colobactin protein, which gets released somehow from the bacteria and either delivered into a mammalian cell to cause cancer, or in our case, into the extracellular space where it can come into contact with Staph aureus. So one important thing about this system is that colobactin, in theory, might be able to kill E. coli itself. And so E. coli then makes a protein, an immunity protein called CLBS, which protects E. coli, binds up colobactin, so colobactin can't kill E. coli. And one of the things I forgot to mention was that the way colobactin kills is that it intercalates into DNA and to cause DNA damage, and that causes this genotoxic effect. So we asked whether this CLBS is protective to Staph aureus from colobactin killing, and that would then tell us that really it's this colobactin molecule that's encoded at this island and not some other intermediate that's important for killing Staph aureus. And so indeed, when we express CLBS in Staph aureus, it is protective against E. coli mediated killing. Here again is single species Staph aureus biofilms. If we mix with E. coli controls, it is readily killed. But if we mix with a Staph aureus expressing CLBS, it is protected. So this really tells us that it's that colobactin molecule from E. coli that's killing Staph aureus. So now let me move to this BAR-A UVRY system. And so what this is a BAR-A is a membrane-bound sensor molecule that then, when activated, can communicate with a cytoplasmic transcriptional regulatory protein called UVRY to cause transcription of genes that are downstream. And so this had never been associated with PKS regulation before, except in one thesis, PhD thesis that we found online that was written in German, but we Google translated it. And what we found was that there was this one report that some E. coli strains that had natural mutations in the UVRY gene lost their cytopathic effects on human cells. And so there was this idea that maybe this sensing system, this regulatory system, might be involved in PKS regulation. But that was interesting to us because many people had studied PKS regulation because there's much interest in it. And it had always been associated with iron. So under low iron conditions, PKS transcription is induced, and under high iron conditions, it is repressed. And so this makes sense in the mammalian host. Iron is restricted. This would get turned on. And so this would make sense in the context of human colorectal cancer. But in work I'm not showing you, we really could eliminate the possibility that iron was controlling PKS in this mixed species interaction. So then this left us, one other thing before I go on, and that's despite the fact that we know that these bacteria, when in mixed species interactions, are starved for iron. They're competing for the limited iron in the growth media. And we know that because both species in mixed community biofilms activate genes that are involved in iron acquisition. So it says that the bacteria are iron starved. They're trying to upregulate iron acquisition machinery. So they are iron starved, but yet this does not seem to be the regulatory mechanism for PKS production in these mixed species biofilms. So this left us with the hypothesis that the BAR-A system is involved in regulation. So then a little bit more about how this works. So BAR-A senses small molecules in the environment. When those molecules are active, it becomes activated, activates UVRY, transcription of downstream genes. And one of the important ones is this CSRB, which encodes a small regulatory RNA that binds to a global regulator protein called CSRA. So when CSRB is made, it binds up CSRA, and CSRA can't do its job. And what CSRA does is it positively regulates glycolysis and certain types of metabolism, and it also positively regulates motility and virulence. And these are basically usually thought to be non-biofilm phenotypes. And it, at the same time, suppresses different types of metabolism and biofilm formation. And we postulated then that it would also be involved in suppressing production of colobactin. And so I'm going to just summarize a lot of work in this little cartoon, and to tell you that this is actually true, because if we get rid of BAR-A and UVRY, those mutants, we then lose the production of CSRB, and then therefore we can't have CSRA sequestered, which means we have an abundance of CSRA present. And what that then means is that we have more suppression of all of these phenotypes or activities, including colobactin production. And using genetic approaches, we can indeed show that if we manipulate this CSR system in this way, we see reduction of colobactin production and reduction of Staph aureus killing. So this PKS island is definitely controlled by this BAR-A UVRY system in E. coli in these mixed species biofilms. So this was exciting for us. The other thing that's really interesting is that we can see that the genes encoding the colobactin biosynthesis machinery are significantly increased in transcription in the presence of Staph aureus, shown in blue, compared to the gray single species biofilms. So this says that E. coli is sensing Staph aureus and turning on PKS production. And what we know about the molecules that turn on this BAR-A UVRY system is that things like formate, lactate, short-chain fatty acids can be sensed by this system and turn it on. And what's cool is that we know from the literature that iron starvation, like what would be occurring both in a mammalian host or during this mixed species biofilm, as I just showed you, iron starvation leads to increased production by Staph aureus of lactate, formate, and acetate. And in E. coli, iron limitation also leads to the accumulation of these same molecules. So we think in these mixed species biofilms, these BAR-A activator molecules are being accumulated, which allow us then to come to our final model. Again, in low iron conditions, either of mixed species competition for nutrient iron or in the mammalian host, Both E. coli and Staph aureus are producing these short-chain fatty acids that accumulate in biofilm settings at high concentrations, which trigger E. coli through the BARA-UVRY system to produce this genotoxin called colibactin, which we know then can be specifically acting on Staph aureus to cause DNA damage and kill Staph aureus. So I've given you a either very brief or a little bit more detailed summary of how we think about these mixed species interactions during biofilm communities. These principles could be applied to any two species of interest that you study in the oral cavity just as readily as we've done here for these wound infections. And so with that, I will stop. My time is up. So I pointed out the people that did the work already. So again, a thank you to them, to our collaborators, our funding here in Singapore. Thank you for listening, and I'll look forward to the session later where we can have a more discussion about this.
Okay, thank you very much, Professor Khan, for your inspiring and very excellent speech. So this is a kind of reminder from chairperson. The audience may write down your question. You don't have to until the speech is the end of the speech. So you can write down anytime and we will collect all the questions and we can ask Professor Kimberly later, okay? Thank you very much. So, so Professor Waruna, is Professor Noor Adina here online already?
Yes, she's here.
Okay, okay, thank you very much.
Dr. Adina, are you here?
Yes, I'm here. I'm ready.
Yes, you can share your screen and start the talk.
Let me just start now.
Okay, sorry, I will introduce Associate Professor Noor Adina. And Professor Noor Adina received her PhD of clinical dentistry in 2010 from University of Otago, New Zealand. And she continues her research in the field of regeneration, animal model, and obesity related to periodontal disease. She has published excellent articles in various international journals. So let's welcome Professor Noor Adina Baharuddin. Thank you very much. You may share your slide.
Thank you, Madam Chairperson. Good morning, everyone. Can I get confirmation that you can see my slide?
Yes.
Yes.
Okay, that's great. Okay. Good morning, everyone. Thank you to the IADR committee for this opportunity, for the invitation for me to share my experience at this platform. I'm Associate Professor Dr. Noor Adina Baharuddin from University Malaya. Let me see whether my screen can, okay, yeah, it's moving. Okay, I'm a dentist by training. I specialize in periodontics. I'm currently based at the University Malaya, which is located in Kuala Lumpur. My menu for the presentation is as on the screen. Over the last few decades, there are increasing evidences that associate periodontal disease and systemic diseases. Among the diseases are the type 2 diabetes mellitus, CVD, cardiovascular disease, obesity, and rheumatoid arthritis. For today, we are focusing on the link between obesity and periodontal disease. Literature shows that severity of periodontal disease may be exacerbated by some conditions associated with obesity, including hypertension and insulin resistance. Conversely, active periodontal disease may exaggerate systemic inflammation thereby increase the risk for chronic inflammation, chronic diseases. This multi-directional relationship between periodontal disease, obesity, and obesity-related chronic diseases could be attributed to the fact that these conditions share the same pathophysiological pathway. Just some quick facts on periodontal disease to set the stage. Periodontal disease is multifactorial disease with the dental biofilm as the initiator. The host immune response against the bacteria present in the dental biofilm causes inflammation. Hence, the dental biofilm initiates a disease process, which causes destructions of the supporting structures of the teeth. There are two common ones. The first one is gingivitis, a milder form of periodontal disease. It's self-limiting and reversible. What I meant by reversible is the ability to bring back to healthy periodontium once the dental biofilm, the plaque, is back under control. And what I meant by the other one is periodontitis, which is the advanced form of periodontal disease. In periodontitis, the host-mediated reaction resulted in deepening of periodontal pockets, and thus the subgingival plaque migrates apically, perpetuates the inflammatory reaction, and at the end, the alveolar bone resorption takes place. It is a major cause of tooth loss. It may affect your speech. It causes low self-esteem and resulted in a poorer quality of life. Periodontitis is a major concern. It is reported as the sixth most prevalent oral condition worldwide. It's a major contributor to the global burden of disease. It has a prevalence of 30 to 35% worldwide, and approximately 11% of the global adult population suffers from the severe type of periodontitis. In Malaysia, the prevalence is reported as 48%, and almost half of those affected belong to the severe group. A bit about obesity. Obesity is a condition in which abnormal or excessive fat is deposited in the body, resulting from an imbalance between the intake and usage of food, measured in equivalence of calories. It is primarily attributed to sedentary lifestyle, combination of reduced physical activity and a diet high in fat. But now it becomes concentrated among the poorer community. It is acknowledged that obesity increases the risk for non-communicable diseases such as periodontitis. The important thing about obesity is the adipose tissue. The adipose tissue is involved in metabolic and physiological pathways, such as inflammation. The cells in the adipose tissue release or secrete cytokines and biomarkers, such as TNF-alpha, interleukin-6, CRP protein, and the resistin. And these become responsible for the abnormal levels of inflammatory cytokines found in the obese. The obesity is regarded as a state of low-grade systemic inflammatory condition, which may mediate inflammatory processes in humans, such as periodontitis. Okay, some facts about the prevalence of obesity from WHO. The world prevalence of obesity has tripled since 1975. In 2006, the data showed that 13% of the adults population worldwide were obese, and these accounted for 650 million adults worldwide who are obese, and 340 million children and adolescents between 5 to 19 years old were either obese or overweight. Sadly, most of the world's population live in countries where obesity and overweight kill more people than the underweight. Okay, the next question is, how are they related? Yeah, I share here the proposed model by Jenko 2005, a model linking inflammation to obesity and periodontal disease. Obesity is a significant predictor for periodontal disease. Insulin resistance appears to mediate the relationship. What happened is obesity is associated with high level of TNF-alpha and insoluble receptors, which may in turn lead to hyperinflammatory state responsible for insulin resistance. Together, the diabetes type 2 and decrease in insulin sensitivity trigger further release of inflammatory cytokines and predispose towards periodontitis. Over the years, there are many studies investigated the relationship between obesity and periodontal disease. It was first reported in experimental animals some 44 years ago. And Saito was the first in 1998 to report the association in human on a healthy Japanese population. It was and subsequently, there's a lot of clinical trials that that been conducted in human to show the in different population to show the association. Among all was Kadi et al. In the Jordanian obese population. He reported that obese has a higher risk to develop periodontal disease, has higher risk for periodontal disease. Hunt et al. This is the Malaysian study, sorry. In addition to, he showed that 74% of the obese presented with periodontitis. And he also showed that there is no difference in terms of oral health quality of life between the two groups. Yeah. Al-Zahrani in 2003 used the third enhanced data. He used the regression models to estimate the association. It was reported that younger people are associated with increased periodontal disease prevalence, and the odds ratio for periodontitis is 1.76 if you're obese. I share here a few, before that. Another human studies, Morita in 2011 did a longitudinal studies. They evaluated the BMI towards the periodontal disease development. They observed the BMI at baseline and 5 years. And what they found is that there is a positive relationship between BMI and periodontal disease. They reported that the hazard ratio for developing periodontal disease is much higher for the obese. And there is a dose dependent between BMI and development of periodontal disease. I shared systematic review and meta-analysis here. If you see here, Cafe and Survan in 2010 and 2011 consistently showed positive association between obesity and periodontal disease. However, they commented that there's limited longitudinal studies that's unable to distinguish the temporal order of the events, meaning that they can't actually dictate whether it is the obesity that leads to periodontitis or the other way around. Meta-analysis by Maura Gregg in 2014 meta-analysis by Maura Gregg in 2014 showed a significant association between obesity and periodontitis, and they highlighted that risk factor should be better clarified to explain the direction of the relationship. So, despite many research and resources spent, we are still not clear as to what is the mechanism that link obesity and periodontal disease. Our team at University of Malaya also explored on obesity and periodontal disease. We have looked into the prevalence both in adults and adolescents, as well as in the intervention part. We believe that obesity and periodontitis are related. What I would like to share next is part of research conducted at University of Malaya. This was done about a few years ago and nothing outstanding. It's just a manuscript that's being on my desk for some time. The issue here is I have the results, but I don't have the answers that I'm looking for. I wish to know more about the mechanism of the link, if not the mechanisms itself. Briefly about the study, we did a comparative study aimed to quantify serum resistant and periodontal pathogens in Malaysian obese subjects with periodontitis and correlate their levels with clinical parameters. We used the case definition by AK for periodontitis. The inclusion and exclusion criteria were as on the screen. Sorry. Figure 1 summarized the study design. Basically, once we recruited and based on the inclusion and exclusion criteria, we look at the biometric measurement and decide whether they are obese or the normal weight. We did the questionnaire and full mouth examination, collect the samples, and run the laboratory analysis. So I would like to highlight important things in the study for reflection purposes. So table 1 summarized the subject's characteristics for the study. We can see that there is significant differences for age, gender, ethnicity, smoking status, and education level between the normal weight and obese groups. Remember, these are all periodontitis patients. Yeah? In table 2, we look at the comparisons between the clinical parameters in the two groups. What we can say is the obese group had higher plaque and higher bleeding index, indicating inflammation. On the, we segregate the clinical parameters in terms of pocketing based on the severity of the disease. If you look at the pocket depth, if it's less than 4 means it's healthy, 4 to 6 and more than 6 indicated more severe periodontal condition. So what we found is in a normal weight group, they have more sites with severe diseases. In this case, it's about 30%, mean percentage of sites with pockets more than 4 millimeters, as compared to about 17% in the obese group. All right, I would like to bring your attention to this slide. In terms of serum resistance, we found that the serum level of resistance is significantly higher in the obese compared to the normal weight group. The increase in the serum resistance level could be attributed by number 1, the systemic effects of obesity. The obese adipose tissue exhibited larger adipocytes and more macrophages, thus resulted in greater production of resistin. And number 2, from the local periodontal inflammation, periodontal pathogens might stimulate adipose tissue and macrophages to produce more resistin. So being obese, it is expected that you will have more of these cytokines in the system. And it was suggested that it's possible that the local inflammation may modulate or regulate the systemic level of resistin. The findings are supported by other literatures. We found that periodontitis patients had higher serum resistant levels compared to the healthy people. And this is in line with the increased local pro-inflammatory cytokines associated with elevated localized inflammatory burden. Obese subjects demonstrated higher serum resistant levels compared to the lean subjects, and there have been shown a positive correlation observed between with the BMI changes, visceral fat, and fat content. What it means here that the elevated levels alters the host immune response, meaning that the obese subjects are more susceptible for the incoming bacterial insult than the normal weight. and obesity potentially modulate one's host immune system and increase one's susceptibility towards periodontitis. With regards to the periodontal pathogens, we found that in the study we found that the highest count of PG, the highest count detected was PG, followed by T. forsythia and P. intermedia, T. forsythia and P. intermedia for both groups. And these findings were in agreement with previous studies. It shows that periodontitis subjects it shows that the periodontitis subjects harbored periodontal pathogens responsible for periodontitis. The mean count for PG and TF were significantly higher in the normal weight. This is due to the normal weight. Remember, I showed just now, they had more sites with deeper pockets, more than 4 millimeter. They had about 30% percentage of sites more than 4 millimeters. And they also have deeper periodontal pocket depth. This in line with observation by Hapaji and Sokransky, whereby higher count of periodontal pathogens were observed in severe disease. We run the correlation between clinical parameters and periodontal pathogens in the subject population. We found that in normal and the obese subjects, there are negligible correlations between the clinical parameters and the periodontal pathogens. Few things that worth noting. Though the correlations are negligible, this should not be taken lightly, as all the subjects are periodontitis patients. The bacteria here, the PG, TF, and T. denticola here, are considered as etiological agents in the pathogenesis of periodontitis, and their presence in the pocket is linked to the progression of the disease, are considered as strong markers for periodontitis in adults. So overall, the results consistent with the current knowledge and understanding, but provide no better insight as to what we already know. It is possible that we are not walking on the right path. Are we doing the same thing over and over again and expecting different results? Is it possible that we can't pinpoint to the mechanism linking obesity and periodontal disease because we are not clear about the pathogenesis of the disease itself? For that, I would like to revisit the pathobiology of periodontal disease to understand the disease further. It is well established that the innate and adaptive immune systems play a key role in the inflammatory response of periodontal disease. The classic paper by Le et al. On experimental gingivitis is very interesting. They asked the subjects to abstain from oral hygiene measures for 3 weeks, and all subjects showed the plaque accumulation, gingival inflammation, and the inflammation resolved following the institution of the plaque, the mechanical plaque control, meaning that they asked patients not to brush for 3 weeks, and patients developed the disease. And this condition is reversed by asking the subjects to clean their teeth again. However, interestingly, we see that the rate of gingivitis development were inconsistent, meaning that people have different timing over the 3 weeks. Some people have gingivitis at 10 days, some people have at 14 days, some people had it at 21 days. So this brings about the concept about susceptibility of the disease, which is coming from the host response. I think about a few years ago, probably 5-6 years ago, we come up with the dysbiosis concept. Actually, what we know is in health, in clinical health, there is a symbiosis between the microbes and the host response. What happens is clinical health is the balance between the microorganisms in the system and the host. And this symbiosis occurs when the mass of biofilm increase, thus change the microenvironment and its constituent. Because of that, current sensing or the keystone pathogens concept has taken place. Pathogens such as the P. gingivalis influence the whole community of the dysbiotic biofilm, and they exert a greater virulence to the host. Periodontitis is regarded as a dysbiosis disease affecting the tooth supporting structures. I came across this new model proposed recently. This is by Van Dyck and Bartol. Okay, it is called the inflammatory mediated polymicrobial emergence dysbiotic exacerbation, the IMPEDE theory or IMPEDE model. This is actually represent a paradigm shift related to the pathogenesis of the disease. We know that in periodontal disease, inflammations and polymicrobial biofilms are involved. But what is the connection? The connection is the most confusing part. Despite the advance in periodontal microbiology and the issues about which come first, the temporal, the spatial relationship as comparison to the inflammatory response or the changes in this symbiosis is still debatable. The good thing about this model is that it incorporates the health. Previously, our classification diagnosis does not involve health. This model continues from health into 4 stages of disease, and it includes the severity and complexity of the disease, as well as the extent and distribution of the disease. All right, okay, let me explain about the model a bit more. Okay, the model answered 3 major questions. Number 1 is what initiates the disease? They're talking about in health and gingivitis. Okay, what we know in clinical health, there is low-grade inflammation, the presence of neutrophils in the gingiva with no clinical signs of inflammation. Basically, it is referred as homeostasis in human. Failure to disrupt the polymicrobial biofilms leads to accumulation of biofilm at the gingival margin, and it is regarded as gingivitis. Question number 2 is what exacerbates the disease? This is referring to stage 2 and 3 and possibly 4. It is referring at the switching from gingivitis to periodontitis, from a reversible condition to irreversible condition. This is the tricky bit because there is limited data on the associations of the disease, association between the inflammation and the microbial involved, and thus the cause and effects interference are limited. So what they come up what they're referring to for this model is just based on consistent observations. And they found that there is, from observations, there is a change in the subgingival environment is related to chronic inflammation. And with a change in the composition and proportions of the bacterial species in polymicrobial biofilm that characterize dysbiosis. And there is reasonable evidence in other systems, other than the oral systems, that inflammation can cause dysbiosis, and dysbiosis induces inflammation. And they found that it was evidence in other systems that highly unlikely that the bacteria or the host is the sole driver of the disease. However, the exact temporal relationship and contribution between the host and the bacteria is not well described. With this model, they come up with the polymicrobial emergence concept. What it means that when they shift from gingivitis to periodontitis, there is emergence of gram-negative species. And this is normally common with chronic inflammation. And they noted that this comes with a diverse polymicrobial composed of disease-causing species they refer as pathobiont, and the commensal, the same biont species. All subgingival ecological and environmental changes are driven by unresolved chronic inflammation related to host susceptibility and the disease itself. Meaning that if you have a low susceptibility to the disease, then you may be able to contain the condition into gingivitis for indefinite number of years. Yeah? The other thing, the other important thing about the polymicrobial diversity is that the process can be controlled by driving resolution of inflammation, meaning that you want to allow the commensal biofilm to return as the predominant microbiota. The third question is that what resolves the disease? They're talking about the resolution of inflammation, returning back to homeostasis state, and re-establishment of truly commensal plaque microbiome. This is not easy in the periodontitis condition because the fact that there is heavy bacterial load in the mouth, so this resolution does not occur spontaneously. And the fact that periodontitis is a condition where disease and healing occurs simultaneously. And the dysbiosis of subgingival plaque microbiome drive the inflammations, perpetuating the disease further. The conclusion coming from this new impede model or theory is that the current driver is the inflammation itself. Only at a later stage, the microbial specificity, meaning the pathogenicity, starts to play a role. Studies demonstrated that there is an increase in microbial diversity of gingival plaque from health to gingivitis and to periodontitis. However, most studies on microbial diversity at periodontal sites have not differentiated between the severity of the disease associated with deep pockets. Kirst et al. In 2015 tried to differentiate between 6 millimeter and 7 to 8 millimeter pockets. They found that there's higher diversity and species richness in 6 millimeter pockets. What they concluded is the subgingival microbial at disease sites were more homogeneous at disease sites, suggesting that as you go deeper into the pockets, as the pocket gets deeper, there is a limited repertoire of species involved in disease progression. So as a summary, what I can say is that the available evidence are still relevant to date. The impede model enhanced pathobiology of the periodontal disease, yet there is still gap in knowledge due to limited association studies, and there is a need to explore the diversity of microbiome in periodontal disease, more longitudinal or association studies to improve existing knowledge. So coming back to the topic, what is the relation between obesity and periodontal disease? If I were to add the obesity into the equation, into the impede model, what I can say is in terms of exacerbation of the disease, obesity definitely adds burden of inflammation. This has been shown in Jenko hypothesis. And what happened is it will exaggerate the polymicrobial, and it will cause the dysbiosis, which is driven by the inflammation. In terms of resolution, in order to achieve hemostasis and the commensal bacteria, I think periodontal treatment would be unable to control the systemic inflammations contributed by obesity. We may have to look into weight control in order to remove the systemic inflammation from the obesity. Could it be the microbiota? It's a bit complex, and there is a gap in the pathobiology of periodontal disease, and there's limited association studies available, and the spatial and temporal relationship is unclear. So with that, I thank you.
Okay, thank you very much for Professor Noor Adiner for your wonderful speech. So we will have a panel discussion later. This is a kind of reminder from Chairperson. We welcome all the questions, and please write down your question in chat room, and we will collect all the questions and discuss later. So now I'm going to introduce the third speaker, Professor Yuan Chen. It's me. Okay, I came from National Taiwan University, and my research mainly focus on the associations between periodontitis and systemic disease, especially arterial disease, SLE, rheumatic arthritis, and Alzheimer's disease. So today my topic will focus on the associations between periodontitis and Alzheimer's disease. And apologize, I have made a video, so I will play a video, then we can discuss later. Thank you very much. Good morning, ladies and gentlemen. I'm Yuan Chen from National Taiwan University. It's my great honor to present my research at this meeting. I would like to express my great appreciations to IDRC committee. Today, my topic is of serology and oral microbiome. My research mainly focus on the associations between periodontal disease and systemic disease. Today, I would like to discuss this association in Alzheimer's disease. Periodontal disease is a common disease worldwide. Around 20% of population live with periodontal disease. Periodontal disease will lead to bone destruction and tooth loss. Periodontitis comes from bacterial infection, and it further triggers the host immune response. Several risk factors, such as oral hygiene, smoking, stress, and many systemic disease are reported to associate with periodontitis. Alzheimer's disease is well known to be a neurodegenerative disorder, and it is the most common type of dementia. Around 10% of the population worldwide suffer from Alzheimer's disease, and the number is still increasing. From memory loss, change of personality and behavior, Alzheimer's disease could be a very serious problem. How may periodontal disease be associated with Alzheimer's disease? Many studies emphasize the impact of oral microbiome to the whole body. The dysbiosis of oral microbiome is related to oral inflammation and may also contribute to systemic inflammation through bacteremia or other pathway. We all know oral cavity is the first part of digestive tract. Besides, oral microbiome and the fecal microbiome share 45% similarity. Recently, gut-brain axis has been well discussed. They reported the gut microbiome may affect the signal of central nervous system, then affect brain function. So it is reasonable we propose oral brain axis. Oral inflammation might affect brain function through pathways. It has been reported oral treponema and P. gingivalis DNA existed in brain tissues of autopsy of Alzheimer's disease patients. It implies oral bacteria or virus vectors may go through blood-brain barrier and cause damage in brain. There are many reported animal studies. ApoE is the principal cholesterol carrier in the brain. ApoE knockout mice, which are easier to suffer from Alzheimer's disease, showed more P. gingivalis DNA accumulations in brain tissues. In another animal model, when P. gingivalis was injected to gingival margin, increased cerebral A-beta deposition and enhanced intracerebral inflammatory response, as well as a decrease in cognitive function was reported. In addition, interleukin-1 beta and the TNF-alpha levels in the brain were also higher in AD transgenic mice. We can find many kinds of inflammatory cytokines in serum and saliva samples in periodontal disease. However, we also find these cytokines in serum and cerebrospinal fluid in Alzheimer's disease patients. So we would like to ask, will these cytokines associate with or affect each other? Therefore, the aim of this study was to study the associations between periodontal disease and Alzheimer's disease by examining periodontal clinical parameters, serum biomarkers, and oral microbiome. Now, I'm going to introduce the material and methods. Basically, this is a case and control study. The case group is Alzheimer's disease patients, and the control group is healthy elders without Alzheimer's disease. 20 cases and 20 controls were recruited in this study. Age and gender are matched. Alzheimer's disease patients were recruited from Professor Chu, Department of Neurology of National Taiwan University Hospital. The inclusion criteria included pure Alzheimer's disease without mixed type dementia. Age was between 50 to 90 years old. So we only recruited mild Alzheimer's disease patients. Exclusion criteria included severe Alzheimer's disease with MMSE less than 10 points. The reason that we excluded the severe Alzheimer's disease patient was due to their poor cooperation. In addition, the patient who were complete edentulism, full mouth dental implants, lack of posterior support, and who took antibiotics within the 3 months were also excluded. Then, serum and saliva samples were collected, and intraoral examinations were done in both groups. For serum samples, we detected AD-related biomarkers such as A-beta 42, tau protein, phosphorylated tau triglyceride, high sensitivity C-reactive protein, pro-inflammatory cytokines, and antibody to P. gingivalis lipopolysaccharide. Then, intraoral examinations and dental X-ray were taken in order to properly diagnose the severity of periodontitis. The periodontal clinical parameters, including probing depth, clinical attachment label, plaque index, and the gingival index, were recorded. We measured the bone level of posterior teeth from panoramic X-ray. We also recorded the residual tooth number. The diagnosis of periodontitis was based on American Academy of Periodontology classification at 2017. In addition, we detected oral microbiome from saliva samples. After DNA extraction, 16S ribosome DNA V3 to B4 region were amplified by PCR. This is the primer we used. Next generation sequencing was used for genomic sequencing by Illumina MiSeq machine. And the sequencing data were analyzed with WuChai software. These are the statistics methods we used. Basically, we used non-parametric test due to the limited sample size. Again, this is the flowchart of this study. Now we are going to show the results. The participants were age and gender matched. The average age of participants was 74 and 73 years old, respectively. Interestingly, the case group showed significantly lower education level than the control group. However, no significant difference in the severity of periodontitis was found between the two groups. In serum biomarkers, significantly higher tau protein, high sensitivity C-reactive protein, and anti-P. gingivalis lipopolysaccharide antibody were found in case group. There were no significant differences in other biomarkers in the two groups. Then we try to analyze the correlations between these biomarkers and the periodontal parameters. Here, I illustrated some important findings. Significantly positive correlation was found between high sensitivity C-reactive protein and the phosphorylated tau protein, high sensitivity C-reactive protein, and the A-beta 42 protein. However, significant negative correlation was found between high sensitivity C-reactive protein and interleukin 10. We also find that anti-P. gingivalis lipopolysaccharide antibody was positively correlated to phosphorylated tau protein and high sensitivity C-reactive protein. This may imply that periodontal infection was associated with systemic inflammation and Alzheimer's disease-related biomarker. Another interesting finding was in the AD group. The severity of AD, which was shown as CDR score, was positively correlated to the severity of periodontitis, which was shown as CAL. Now we move on to the results of saliva microbiome. This is the relative abundance of each patient in case and the control groups. The main composition of microbiome and the top 10 bacteria were similar in the both groups. This is the rarefaction curve, which run the sequences more than 40,000 times, which has enabled us to identify all the OTU within our saliva samples. When comparing the microbiome composition of the case and the control group, the Venn figure and the MRRP analysis showed no significant difference in the two groups. In PLSDA analysis, we could identify 5 to 6% of the microbiome, which were unique in case and the control groups. This is the heat tree diagram, which displayed the taxonomic distribution of case and control groups. The taxonomic distribution were similar in general. There is another interesting finding. In general level, we find significantly higher Prevotella, Peptococcus, Faecolibacterium, and Noxibacillus in Alzheimer's disease group. And in species level, significantly higher Prevotella baca was found in Alzheimer's disease group. Prevotella baca has been reported to associate with odontogenic infection. Here we come to the discussion part. We find that Alzheimer's disease group with lower education level, which was consistent with the 58% of the studies according to the systemic review by Professor Sharp at 2011. In addition, this study didn't find the significant difference in the severity of periodontitis between case and the control groups, which was inconsistent to the previous study. This may be due to the age was too old in our study population. Both of case and the controls showed certain level of periodontal destruction. For serum data, our results are consistent with previous studies. Professor Schenker found that serum tau protein was higher in AD group than in minor cognitive impairment group. Professor Song also found serum high sensitivity C-reactive protein was higher in the AD group than non-AD group. However, the top 5 oral microbiome was different with the study conducted by Professor Willis at 2018. We noticed that in Willis' study, the participants are Spanish teenagers. From literatures, oral microbiome could change with environment aging, smoking, and life habit. As for no significant difference in oral microbiome between the two groups, the reason might be both groups show similar severity of periodontitis and shared the similar living environment and culture. Here we come to the conclusion. First, the study found that Alzheimer's disease was associated with higher serum, high sensitivity C-reactive protein, tau protein, and anti-P gingivalis lipopolysaccharide antibody. Second, the severity of the dementia was positively correlated to the severity of periodontal destruction. Third, only very few bacterial abundance of oral microbiome was significantly higher in the AD group. Prevotella baca was found significantly more in the AD's saliva by NGS technique. Further studies are warranted to verify the role of Prevotella baca in the pathogenesis of Alzheimer's disease. Thank you very much for your attention and wish you stay safe from COVID-19. Thank you very much. Okay, thank you very much. I think we still have 5 minutes for panel discussion. So just now I have collected the questions. We have 3 questions for Professor Kimberly and also for Professor Noor Adiner. So I will show the slide. Okay, so the first question is from Dr. Prasanna, is for Professor Kimberly.
Should I just read it?
Yeah, okay. Okay, can I read? There is some body of evidence that specific environmental cues in the cultural medium are needed to replicate real-life conditions. So are there any specific guidelines in your group for the optimal cultural medium for different infection models? Sorry.
Yeah. Hi, Prasanna. Thanks. I think that's a really good and important question. And the bottom line is no, I don't have any specific guidelines. We can force any sort of interaction we want by changing around the infection, like the components of the host, the growth media. So I think, it's really important just to think about what infection environment you're modeling, and then go from there. So if we're trying to, model a urinary tract, environment versus a wound environment, we think about different components. But I don't think there's a magic answer to this. There's a lot of trial and error to get this right. This is why we start with the in vivo model, and then we try to build in vitro models that look like it. And even if we build an in vitro model that looks like what we're studying in vivo, we still don't see 100% correlation. But I think it helps us a little bit doing it that way. But yeah, this is just labor intensive to get the right model, I think.
Okay, thank you very much. And we have another question. So it's from me, yeah. So it's very interesting. You mentioned the synergistic effect of E. coli and the Streptococcus aureus and also E. faecalis, these 3 bacteria, and the mechanism may involve ion transfer or glycolysis. Yeah. So I would like to know, have you ever worked on the periodontal bacteria such as P. gingivalis or tenorrhea forsythia or oral trypanosomas. Do you have any good advice if you want to explore their associations, in periodontal disease? Thank you very much.
Yeah, that's also a great question. And no, we have not gone after any interactions that are relevant to the oral cavity. That just hasn't been our focus, but there's no reason that it shouldn't be done. And I hope somebody would do that work. I'd be very happy to collaborate and help with that. I think the best advice for starting this is, if you have either an in vivo, which is hard, I think, or in vitro model that, you think is the best mimic for what's happening in the human oral cavity, I would start with that, and if you know that two species are interacting and one, in disease is more dominant than the other, then try to set up an in vitro model that's really mimics that as a starting point to then try to understand interactions. I think important things to look at are bacterial burdens, but also, bacterial organization within, in proximity to each other. So imaging type experiments. And we've gotten a lot of mileage from transcriptomic type analyses as well, mixed versus single species biofilms. So those might be sort of easy places to start, at least to start to formulate hypotheses to test about the associations.
Okay, thank you very much. Yeah, it's very inspiring. We have a lot to do,
Definitely.
Okay, so I have one question for Professor Noor Adiner. Thank you very much for your inspiring lecture. We understand the association between periodontal disease and obesity mainly come from TNF-alpha. So I just want to know, in your research, have you ever find the dysbiosis of polymicrobiome related to higher TNF-alpha? Because in your lecture, you emphasize the importance and also the information caused by dysbiosis of polymicrobiome. So I just wonder, have you ever checked any kind of inflammatory cytokine elevated in your patient? Thank you.
Thank you for your questions, Prof. Chen. We have looked into the periodontitis and obesity. That was way ago. I think in 2000. I think we started in 2015, and the idea about the dysbiosis is quite recent, probably at the same time it happened. To be honest, that I have not found dysbiosis of the polymicrobiome related to higher TNF alpha in the study.
Okay, so do you ever find other inflammatory cytokines such as interleukin-6 or?
We did when we did the study, mainly we were focusing at resistance at that point in time, because resistance was the at that point in time the new cytokines of interest. The rest are kind of settled and well established. So that is why we are looking at the resistance mainly.
Okay, I see. Thank you very much. Thank you. So the last question is for me. Yeah. So I will just read. Okay. The question is, do you think the difference of microbiome between two groups because of the cognitive difference? Oral healthy of Alzheimer's disease is poorer than non-Alzheimer's disease. Okay, so thank you very much for Mr. Tang Fang. Okay, my answer is because in my study population, both of them, like Alzheimer's disease patient and also the non-Alzheimer's disease patient, actually their oral hygiene is similar, and also their prey index similar. So their periodontal status were also similar. This may be because their age were so old, because they are like more than 70 years old to 90 years old. So they have certain kind of destruction of periodontal disease. So in my study, I couldn't find the periodontal disease in Alzheimer's disease is more severe because of the age. Okay, and the other question is also to me. I will read the question. In case of periodontal disease associated with systemic disease like obesity, sorry, it's for Professor Noor Adiner. It's okay. I will read the question again. In case of periodontal disease associated with systemic disease like obesity, do we need to address first managing at the medical condition level, or can we proceed right away to treating the periodontitis? It's from Dr. Joseph Acosta.
All right, okay. Regarding this periodontitis and obesity, or any systemic diseases, it depends on what kind of systemic diseases that we are referring to. In cases like diabetes mellitus, where any what you call uncontrolled diabetes could have an impact into the periodontitis management, we have to address the periodontitis, the diabetes mellitus first. In other condition, for example, if you're talking about obesity, it does not have so much of implications if you were to treat the periodontitis first. But nevertheless, how I wish that I can get these patients who are obese with periodontitis for weight management so that we can see the impact. Because if you were just to treat periodontal disease itself, it will not have any significant improvement in terms of the disease itself. Thank you.
Okay, thank you very much. So since we are running out of time, so before closing this symposium, I would like to thank Professor Kimberly and Professor Noor Adiner and Professor Urana for your contributions to this symposium and for making this symposium so successful. So I would like to close this symposium, and we look forward to seeing you next year.
Thank you, Prof.
Okay, thank you very much. Thank you, Professor Kimberly and Professor Noor Adiner.
Thank you. Nice meeting you.
Thank you.
Thank you very much. Thank
You
you.
can leave the hall.
Yes. I think, thank you, Professor Chen.
Thank you, Professor Urana. Thank you very much.
Thank you,
very
Professor
much.
Chen. Yeah, it was very nice. You have done, you have prepared a lot to do a very nice symposium.
Thank you very much. Sorry, yeah, it's my first time. Yeah, thank you very much for your invitation.
Yeah.
Okay, and please transfer my best regards to Professor May.
Yeah,
May
sure, I will. I think she's busy with
I-
some other work.
Yeah, I learned a lot from her. Yeah, thank you very much. Yeah, thank you very much for a great job. Okay, thank
Yeah.
you.
Thank you.
See
Prof.
you.
See you.
Bye-bye.
Bye.
这边。
对，可是因为刚刚好像来不及调，刚刚就是老师还是维持在麦克风，所以刚刚老师一关掉，这边马上就没有声音了，所以我才请老师赶快开起来。对，所以是这边要选择说要与系统相同，那就可以关掉，系统就会有声音。
好，
对。
Hello, Sriram.
Hello, hi Varuna.
How are you?
Yeah, fine. Is it cold over there?
Yeah, it's the so-called winter here. Not that cold, but yeah, it's around 16, 17.
Okay. All right, yeah. So how is the conference going on till now?
I think it's going okay
Yeah,
so far.
yeah, yes. I was going through other sessions. It's quite nice.
Yeah, yeah.
And the platform is also quite good.
Yes, it looks quite nice.
Right. Maybe like should we test?
Yes, we can check our screen share.
Yeah. Is it in the screen? Is it in the screen show mode?
Yeah, I can see your screen.
So you can see the title slide, or is it in the PowerPoint?
Yeah, I can see the full screen somehow. I mean, the full slide.
Varuna?
Yeah.
Varuna, right now in hall number 1, we are having the keynote lecture. That's going to finish in about 4-5 minutes. So then we will live stream it in the same hall, this symposium.
Oh, okay, okay.
So we'll have to wait for 4-5 minutes. They're just wrapping it up. So as soon as they finish, we will then, live stream you there. So just give us about 4-5 minutes.
Okay.
Okay. Yeah.
I think we need to mute
Hello, Varuna? Hello, Varuna.
Yes. Hi, Bharat.
Varuna, you can now the keynote is over. We are streaming you live, so you can start your session.
Okay, so we can stay in the same room, right?
Yeah. You can stay in the same room. You don't have to go anywhere. You can start your session now.
Oh, okay.
Okay.
Can you see my screen?
Yes.
Can you see my screen?
Yes. I got some feedback. Is that okay now?
It seems so.
Is that okay?
Yes, I think so.
Okay, then maybe I can introduce the speaker now.
But still, audio is not very clear.
You don't hear me well?
No, now it's okay. Yes.
Okay, maybe I can start. I can introduce the speakers and then we can start.
Okay, I think that we are still waiting for 2 speakers, but I'm not sure whether they are going to log in now.
Yeah, but I think we should start. Yeah. It's already live. I think.
Yes.
Yeah, so the first speaker is here already, right? So I can introduce just the speakers. So welcome all to our Young Scientist Forum. There are actually 7 young scientists, so we break it down into 2 halls. And in this hall, we are having talks from 4 young scientists. I would like to briefly introduce the different speakers. Our first speaker is Dr. Saran Gopal from National University of Singapore. So Dr. Gopal is an assistant professor at the University Faculty of Dentistry, National University of Singapore. So he is also a thrust co-lead for additive manufacturing for craniofacial and oral applicants at the NUS Center for Additive Manufacturing.
His current research is centered on oral and craniofacial tissue regeneration through the amalgamation of complementary and multidisciplinary tissue engineering technology. Our second speaker is Dr. Varuna Disananaika. So Dr. Varuna is an assistant professor at the Faculty of Dentistry, University of Hong Kong, and his research is currently focused on enhancing post-implantation stem cell survival and vascularization during tissue regeneration, understanding the molecular mechanisms behind the dental stem cells, intercellular cross-link and lineage-specific differentiation on dental stem cell into odontal, osteoblast, and endothelial cells. The third speaker is Dr. Han Kong Nam. Dr. Lam is a lecturer and researcher in the Laboratory of Oral Maxillofacial Biology at the Faculty of Stomatology, University of Medicine and Pharmacy at Ho Chi Minh City. His research interests includes single-cell, osteoimmunology, big data in dentistry, bone regeneration, oral biology. And the last but not least, our speaker is Dr. Sitra from Grantia Violari. Dr. Sitra obtained her PhD degree from the Health Science University of Hokkaido, Japan in 2018. So right now he's working as a lecturer at the Department of Oral Biology, Faculty of Dentistry in the University of Indonesia. Her research interest is oral microbiology, and which focus on oral microbiome study and oral biomarker study in non-communicable disease. So today our chair is Dr. Varuna. So may I pass the time to Dr. Varuna to chair the session. Thank you.
Thank you, Professor May Wong, for your introductions. So our first speaker is Dr. Sriram. And before we start, I would like to remind the audience that you can post all the questions using the Ask a Question button on the platform. So you don't need to wait until the presentations are over. So you can post your questions at any time and we can answer the questions once the presentations are over. So Dr. Sriram, I would like to invite you to start your presentation first.
Right. You're able to see the screen?
Yes. Yes.
All right. Yeah.
We can.
Yeah. Thank you, to Professor May Wong and Dr. Varuna for your kind introduction and this opportunity to present in front of this panel for organized by IDEA Southeast Asia and University of Hong Kong. So in this talk, what I would be covering is about the various tools, next generation tools or multidisciplinary tools which can be utilized for biofabrication of gingival tissue constructs. Right. So starting with traditionally all the research has been done using monolayer cultures of say keratinocytes, fibroblasts, or any other cell types, right? Like so in terms of interaction with other, like let us say host microbial interaction or host biomaterial interactions and so on. But this is something like which is very two-dimensional, and in the body, everything exists in three-dimensional environment with a lot of interactions in three-dimensional space with the surrounding cells and the matrix, right? So that's where like now the research is primarily focusing on how can we like recapitulate the 3D microenvironment in vitro and also move away from animal models because of the interspecies differences and so on. So that's where like the first technology which researchers started to work on is cell aggregates, also called as spheroids or organoids. And then from there to organotypic cultures of multiple cell types, not just one single cell type. So by organoids, what we mean here is three-dimensional, like a ball of cells, where like multiple cells interact with each other. Right. So from there, like people started to move into organotypic cultures. And this is something very, very commonly used, especially for barrier tissues like the skin, the gut, or the oral mucosa, right? And even including the lung tissues. So these barrier tissues are quite commonly like cultured using these organotypic cultures. So that's something like I will cover through this talk as well. So, and then from the organotypic cultures, what has been like going on in the recent, let us say like 5 to 6 years, is the utilization of technologies like 3D bioprinters or 3D biomaterial printers to have a spatial control in terms of what can be, how you can have a geometric control of the tissue to be regenerated. And another aspect where focus is also is on microfluidic tissue microfluidic organ-on-chip platforms, which are basically like a platform which helps to mimic the blood supply, right? And to aid this, there are a lot of developments happening in the imaging front as well, which helps to image three-dimensional tissues in live without need for it to be fixed and processed, right? And seen at two-dimensional cross-sectional views, right? Which is normally done in histological processing. So I will sort of like cover in this talk like these different technological tools, how it can be utilized for gingival tissue regeneration, right? So I would first like start with like how like tissue engineered gingiva can be used for various applications. Like in the academic research, it can be used as a basic research tool to understand the whole biology of the oral mucosa or the gingival tissues and its interaction with surrounding environment, which then brings down to the utilization for consumer care products like toothpaste or mouthwash and other even dental materials. Right. So, and from there are also applications in the pharmaceutical industry where like now there are a lot of these technologies which have been developed for drug delivery using patches or creams through which oral mucosa can be used as a platform to deliver drugs for systemic applications. Right. So these are from a industrial or a more translational perspective. And of course, from a clinical perspective, like it is also essential applicable for use as a tissue grafts, right? So for this, for various these applications, like how can we tissue engineer gingiva, is by mimicking the various cell types which are used. And if you really see the gingival tissues, it is composed of epithelium on the top, which is primarily like composed of keratinocytes. And down below, we have the lamina propria and the submucosa, which is composed of fibroblasts as the primary cells, along with blood vessel cells, which are formed by the endothelial cells, and the surrounding extracellular matrix. So it's essential, if we have to tissue engineer gingival constructs in vitro, we need to consider the integration of these different cell types, how they interact with each other, what is the media conditions which are required for them, and how they interact with the surrounding extracellular matrix as well. Right, so that is where like now like moving from traditional 3D cultures, there has been effort on using flow systems like microfluidics and on spatial control using 3D printers. And like especially the flow system sort of mimics the flow of nutrients or the drainage of metabolic waste, like a simple function of a blood vessel, right? Like so microfluidics helps in mimicking this, while 3D printers help to mimic the spatial control of the cells and the matrix, which are required for the tissue architecture. when we start to like what are these tissue engineered models, like it starts with very simple models called reconstructed oral mucosal epithelium, which is basically the keratinocytes which are grown on these porous culture inserts at air liquid interface. And what happens is that like single layer of keratinocytes stratify themselves one over the other and forms these multi-layered epithelium of the gingiva, the oral mucosa. So these are called as reconstructed oral mucosal epithelium and consist basically of just keratinocytes only. And so it's very simplistic model. And these are commercially available and are used by regulatory agencies for testing of personal care products like mouthwash and toothpaste, right? And moving from still more ahead is something called as the full thickness skin or oral mucosa equivalence, wherein the matrix and fibroblasts are essentially mixed together and placed within these porous culture inserts. And then keratinocytes are seeded on top. And typically what is used is collagen-based matrices, and collagen-based matrices are known for its contraction, as you can see here, like a mountain, right? Like, so this green part is a matrix and with the fibroblast inside, and the top a single layer of keratinocytes being laid down. And then once, when you culture them at air liquid interface or something like a moist air, mimicking the oral cavity or the external environment, the keratinocytes start to form multiple layers, forming a full thickness gingival epithelium. And with this, like opportunities come in terms of adding multiple cell types like endothelial cells for blood vessels, immune cells to mimic the defense system, and including nerve cells. So there are like a lot of researchers working on developing vascularized and innovative tissue models. Right. So essentially what this helps is to recapitulate the stratification and then the cell-cell interactions, cell matrix interactions, and the overall three-dimensional microenvironment. However, still like these tissues do not have a flow system of, even if you have to include the endothelial cells, these endothelial cells are just going to be present there. They're not going to have a flow of blood, right? Like, so that's where like you need to work on microfluidic systems, right? So with that, like we have these various models, like which are commercially available for both the reconstructed epithelium only models as well as full thickness oral mucosa models through Episkin and MedTech. So with that introduction on commercial models and the various three-dimensional models, I would like to go into how we can take this even further by incorporating vascularization and then spatial control and as well as blood flow, right? So we started with first asking this question, like if we have to incorporate blood vessels within these tissues, we need to be able to culture these blood vessels. Means first we should be able to form microvascular networks, and these microvascular networks have to be stable over long term, right? So what we did is like we used endothelial cells and mural cells, which are either it can be pericytes or fibroblasts. And then we first started with culturing them in floating aggregates, or also called as vascular spheroids. And they were seeded within agri-well plates, which are basically like low attachment plates with tiny micro wells. So within a traditional 12-well plate, we can be each well 400 of these micro wells within them. So which means like you can have 400 spheroids made within a single well. So endothelial cells, which were labeled in green, and pericytes, which are labeled in DS red, they were seeded together within these agri-well plates. And then after 24 hours, they start to, because they cannot attach to the plate, they start to aggregate within themselves, forming a ball of cells, or also called as spheroids. And once they are cultured within low attachment plates, they form like these ball of cells. And when we look at using confocal microscopy, these are again live cells without the need for any fixation. what you can see here is the green endothelial cells being on the outside and the red pericytes being reorganized themselves in the center. So initially when we seed, they're all seeded at, in random, but mixed together and seeded in random. But within 24 to 48 hours, they reorganize themselves in such a way that the endothelial cells are placed on the outside and the mural cells, they go to the core of the spheroid. So this is something like an inside-out orientation of a blood vessel, as if the blood vessel is stripped and turn around. The culture media being the lumen of the blood vessel or the blood itself, and the endothelial cells forming the periphery of the spheroids, and the muscular core or the pericytes forming the spheroid core. Right. So with that functionality, then we started to see like whether we can like embed within like a matrix. So these spheroids, they are only composed of cells, right? So there is no matrix involved. But in reality, in three-dimensional tissues, we have a matrix as well. So that's when we started to work on incorporating them within extracellular matrices. So we used a pegylated fibrin gel because collagen gels are known for contraction, and hence it becomes very unpredictable in terms of working with collagen gel. So whereas fibrin gels, on the other hand, is basically derived from blood clot, and so they are naturally angiogenic, and at the same time, they do not have these contractions and inter-experimental variations. Right. So when we started with these pegylated fibrin gels, we included only the endothelial cells, and what you can see is by day 4, they form these nice vascular networks, but by day 6, they just regress. So it's a natural phenomenon which happens in the body, that when there is no support from neural cells like pericytes or vascular smooth muscle cells, the endothelial cells, they just regress. So that's when like we started to work on with incorporating the endothelial cells and pericytes together. And when on day 1, it's basically like you see them together by randomly mixing them together. But eventually these two cells, they self-assemble themselves, forming the vascular endothelial cells. The endothelial cells form these vascular networks, which are stable over this 3-week culture period, as can be seen in this video as well, right? And we also tested with different concentrations of endothelial cells, ranging from 1 to 2.5 million cells for each ml of the matrix. And what we can see here is with more number of endothelial cells, the vascular network parameters keep increasing. So and they reach a sort of a stable point after 2.5 million cells. Beyond that, like the network just like collapses because the whole matrix is eaten away by the cells, right? So this again, like higher magnification confocal images show the lumen formation, and again this video sort of reinforces that like these vascular networks have the endothelial cells have self-assembled to form these microvascular networks, and within them is a hollow lumen, right? So these cells are able to form microvascular networks. So next we asked the question whether they are functional. So one of the functionality of the endothelial cells is to have a barrier properties, right? What is there inside the blood should not leak out to the surrounding tissue fluid, and similarly the other way around, unless like there is a inflammation. So that's where like we use histamine to sort of mimic the inflammatory or allergic reaction, and we used a red colored dye called dextran Texas red, and we did a reverse permeability assay wherein these tissue constructs were cultured with green labeled endothelial cells and pericytes which were not labeled. So that's where like you cannot see the pericytes here. It sort of permeates through the matrix, and if for the cells which for the endothelial cells which are mature, there's hardly any permeation of the dextran Texas red into the lumen of the blood vessels. But if they are treated with histamine, these blood vessels like become permeable, and the dextran-dextrose spread is able to penetrate into the culture medium, into the lumen of the microvascular networks, suggesting the functionality of these microvascular networks. Right. So what we did is like, this is something like done in the manual method where these microvascular networks, they form in a random manner. So next we asked the question, can we control the patterning of the microvasculature using 3D bioprinter or 3D printing? So what we did is like, first we started with developing a bioink, a collagen-based bioink. Collagen and xanthan gum was used. Xanthan gum was used as a thickener so that the collagen is printable. Right. So we started with working in these grid-shaped patterns with different concentrations of collagen and different concentrations of xanthan gum. And as we can see here is that in 8% and 10% xanthan gum with 9 parts of collagen, we have a much more stable printing of these grid patterns. And also these rheological measurements suggest that the collagen bioink has shear thinning properties, which is essential for bioprinting. Right? So that's what we did is we did running using 3D AutoCAD software, wherein like we have 4 layers. So the first layer is just a fibroblast, and the second and the third layer is endothelial cells, which are in crisscross orientation, exactly in 90 degrees to that of the fibroblast layer, and then followed by a final layer of fibroblasts. So it's essentially like a sandwich where endothelials are sandwiched between 2 layers of fibroblasts, right? And so this is a multi-head printhead which can print a fibroblast. One printhead prints the fibroblast, while the other printhead prints the endothelial cells. And what we can see here is over this 6-day period, like these green labeled endothelial cells form these patterned microvascular networks across the whole 3D construct. So at a macro scale, like these endothelial cells seems to form a pattern which is determined by the 3D printer itself. But when we like, and that's something like which we can also see in the scanner view, where the blood vessels are forming only in the grid shape and not in the in between the space, right? So which means like we can control in a larger scale, in the macro scale, the patterning of the microvasculature. But when we really zoom in at each of these strands, what we see is still a self-assembly of these endothelial cells into microvascular networks. So which means at a micro scale, like patterning is still dependent on self-assembly of the endothelial cells within the surrounding bioink or the matrix. So that's where like we are also working with more bioprinter with more finer resolution, so that like we can control the microvasculature patterning even at a finer scale, right? So with that, we next ask this question, like now we can like fabricate this microvascular bed. Can we now like make gingival tissues with on top of this microvascular bed, so that we can have a 3D vascularized gingiva, which can then be like used as a tissue construct for engrafting, right? So what we did is like basically like we took the mucosal matrix, which is fibrin-based matrix, combined it with gingival fibroblasts and microvascular endothelial cells, cultured them for 6 days, followed by seeding of oral keratinocytes and culture at an air-liquid interface, followed for 2 weeks of air-liquid interface culture, right? So these are examples of how these tissues look in a 6-well and a 12-well format. And because the fibrin matrix doesn't shrink, you can see them like occupying the whole tissue insert itself, right, without any contraction. And so this sort of like shows like how the tissue develops over 1, 2, and 3 weeks period, where there is a constant increase in the epithelial thickness and formation of corneal layer, which is needed for the gingival barrier properties of the gingival tissues, right? So So here, like what we see is like higher magnification of the gingival, vascularized gingival construct, where we can see the formation of blood vessels in the matrix. And the immunostaining shows the expression of cytokeratins for the epithelium, and the barrier proteins like the filaggrin and loricrin on the superficial surface. And then the matrix expresses collagen 1 and fibronectin. So remember, like these tissues are made using fibrin, right? So there is no collagen. So whatever collagen is expressed in immunostaining is basically has derived from the cells themselves, like not something like which has been included in the matrix. So other than that, like we also like stain for basement membrane proteins, the laminin 5 and collagen 4, which are expressed at the basement membrane, and also labels all the blood vessels. So here you can clearly see the blood vessels with lumen structures inside, and which also express CD31 and von Willebrand factor. Right. So next, so this is like something like which is done in a traditional manner, where like tissues are fixed after the tissue development happens, and then after that we do sectioning and visualize it at two-dimensional plane, right? So what we started parallelly we were working on is on non-invasive imaging modalities, which can help 3D imaging, so without any use of any labels. So that's where like we started to work on 2-photon microscopy, and which helps to visualize the biomolecules which are present naturally in tissues. And keratinocytes are cells which are very much rich in mitochondria and with a lot of NADPH, nicotine adenine dinucleotide phosphate, right? Like, so these are biomolecules which are present in almost all the cells, and especially on cells which are dependent on energy, right? So keratinocytes have a lot of NADPH, and using that, we sort of optimize like that at around like 740 to 760 nanometer wavelength, excitation wavelength, you have maximum excitation and a more clear visualization of these cells, right? And similarly, the fibroblasts express more of flavin adenine dinucleotide, and hence which are more specifically being more visualizable at 800 nanometers. And similarly, the collagen fibers are visualizable using second harmonic generation, which is a special type of physics principle within the two-photon microscopy, where like, again, only at 860 nanometers, we are able to see the collagen fibers and not in other excitation wavelengths. So just suggesting that like each of these wavelengths are specific to different molecules or different collagen fibers, for instance here, and hence we can utilize this to visualize the different cells and the matrix without using any labels. So this is a sort of montage where you can see the different layers of the epithelium and the collagen in the matrix below, and with the embedded fibroblast. So here in this video, again, like we can see here, the red labeled fibroblast in the matrix, and the epithelium, nothing is there. And then, so this is a combination of all the three colors, where like you have the keratinocytes on the top and collagen fibers in yellow with red colored fibroblasts in the middle. So we can like sort of visualize them in 3D. So we call this as optical biopsy with in three-dimensional space without need. And these are again live tissues. So this sort of opens up opportunities to investigate three-dimensional tissues in live, where like we can study how the tissues change over time or in reaction to any other interventions, right? So with that, next we ask this question, like, can we can form vascularized gingival tissues with blood vessels inside, right? But then there's no blood flowing through it. So that's when we started to ask this question, can we biomimic blood flow? And we started to explore microfluidic organ-on-chip systems, which enable this flow system. right? So a brief introduction on what these organ-on-chip devices are. So these are basically engineered devices which are designed using AutoCAD or three-dimensional tooling softwares, which help to design the various microchannels through which you can flow the culture media or drain the culture media, and as well as micro chambers in which the cells can be cultured. So what this sort of like gives is that like the microchannels help to provide a fluid flow, mimicking the blood flow, and as well as providing a shear stress for the cells to interact and mimic the shear stress which happens in the body, right? And because like there can be like a flow of nutrients on a continuous basis and also in parallel drainage of metabolic waste, which that means like we can have a long-term culture without being worried about the nutrition being used up by the cells, and hence a higher viability and higher performance of the cells. And these all again at the benefit of having low fluid volumes, because everything is at a micro scale. Like, so we have microfluidic channels which are in micrometer scale. So which means like we require very low fluid volumes, right? In addition to that, like it is also possible to, because everything is in micro scale, it is also possible to have real-time monitoring using traditional microscopic tools, as well as integration with biosensors. And because the fluid is continuously flowing, you can also have automated cultures so that the researchers can have probably a good weekend. And eventually all this sort of helps to recapitulate the human physiology much more closer and move away from animal studies. Right. So what we did is like we started off with working this particular chip design where we have like different layers. And each of these layers have different features, like where we have like in the lowermost layer, we have these micro channels with a micro chamber for flow of fluid with a porous micromembrane separating the culture chamber. In the middle chamber is where the cells are cultured. And then above that is that of, again, a micro chamber with micro channels, so that through which we can flow the culture media or air or saliva or even bacteria or any other environmental tools which the cells would interact with, right? And on top is a lid through which we can access. So this is an assembled chip where it's basically like a credit card sized device, like which has four micro chambers in which we can culture the oral mucosa equivalents. Right, so and this is sort of the fluid circuit where like if we were to take a cross section here, this is how it would look like, where we have a micro chamber in the middle and micro channels on the top and bottom, through which using a peristaltic pump media can be continuously perfused through and collected. So this sort of like continuous flow through helps to mimic blood flow and at the same time helps to collect the perfusate, through which we can have a real-time monitoring of various biomarkers which these gingival tissues are expressing. Right, so the workflow is something like we have these microchannels on bottom and the top, the media being flown on the bottom and air on the top, and the matrix with oral fibroblast and oral keratinocytes in the middle. Right, like so we start with only the fibroblast and mucosal matrix, and the media keeps flowing through all through on both the sides, and then keratinocytes are seeded, and after keratinocyte seeding is done, the media flows only through the bottom channel while air is passing through on the top channel, right, which has a moist air, sort of mimic the oral the moist environment within the oral cavity. Right, and then eventually with 2 weeks of this interface culture, we have the formation of gingival tissues, right, under continuous flow conditions, which then can be used for any downstream assays. Right, so this particular slide here like shows like the comparison of the oral mucosa which is developed within a flow system and that of a static traditional two-dimensional traditional insert cultures, right, where there is no flow. And what you can see here is that the tissues which are developed under flow have a much thicker epithelium compared to that of static tissues, and have a much better organization of the basal cells. Higher expression of barrier proteins like the loricrin and filaggrin are much strongly expressed in the flow system compared to the static system, right? And so this sort of like shows how the flow system can enable like mimicking a natural environment, and hence development of much more closer tissue, right? So parallelly, what we also did was like to develop a disease model using an oral ulcer on chip, where the keratinocytes were sort of like removed, where only the fibrin matrix and oral fibroblasts are included. And here, like you can see the vimentin positive fibroblasts in the matrix, and the matrix expressing collagen 1 and fibronectin without any epithelium on top. Right. So next, what we did is like we saw like how we can utilize this to study effect of biomaterials like or oral care products like mouthwash. And because the fluidic system enables flow of any biomaterial, like here, like we were able to flow the mouthwash and help mimic the rinsing of the mouthwash on top of the oral mucosal tissue. So we have the gingival tissue in the middle, and the culture media flows in the bottom, and the test chemical here is the mouthwash. The mouthwash flows on top. So because the fluidics can be controlled, we can have the flow which is in the forward and reverse direction, so help mimic the rinsing of the mouthwash. While the culture media flows always in one direction, but the mouthwash flows in to and fro direction, right? So what we did is like we used two different mouthwash, alcohol-free and alcohol-based mouthwash. PBS was used as negative control, and sodium lauryl sulfate, which is normally used as a detergent within toothpaste and mouthwash, as a positive control. And what we can see here is like the oral mucosa on chip. The negative control, everything seems normal, like there is no cell death. Whereas in the positive control, we have these tunnel positive cells in the epithelium and in the connective tissue, and the epithelium is all disrupted. Whereas in the alcohol-free and alcohol-based mouthwash, the epithelium is able to have a barrier property, and hence prevent epithelial cells being undergoing a cell death. Whereas in that of oral ulcer on chip, we can see the cell death happening in the alcohol-free and alcohol-based mouthwash in the tissue, because of the absence of the overlying epithelium, which helps to form a barrier, right? So this sort of like shows like provides us a model to study how different mouthwashes can be used to study the biocompatibility of the mouthwash in healthy as well as in disease states. So this again like sort of reinforces that using a viability assay, using MTT and on the tissues, and LDH assay on the media which is being collected through the outlet channel, what we can see is that the tissues are viable in alcohol-free and alcohol-based mouthwash, whereas in the ulcer on chip, what we can see is like a higher expression of LDH, suggesting a certain amount of cytotoxicity, which is like sort of threefold increase in the amount of LDH release. So this sort of corresponds to the tunnel assay which was shown in the previous slide, right? So next, we also like started to work on drug delivery and permeation studies, right? So wherein like what we did is like we tested on lidocaine and articaine, where we can see that the lidocaine permeates more faster through oral ulcer on chip compared to that of intact oral mucosa, suggesting that again the barrier properties, and also as a model tool to study drug permeation, right? So with that sort of like sort of like demonstrates how the various tissue engineering technologies can be utilized to have next generation tissue constructs of gingival tissues, right? So with that, I would like to also thank the collaborators from Faculty of Engineering, the Singapore Institute of Manufacturing Technology, the funding agencies from Additive Manufacturing, as well as A-STAR and MOE Singapore. With that, happy to take questions. Thank you.
Thank you, Dr. Sriram, for your very exciting talk. Because of the time limitation, I think we can move on and take the questions at the end of the session.
Yeah, I'm sorry, I didn't like realize the time here in the full screen mode.
Yeah, sure. So yeah, so next, so it's my talk. So I hope you can see my screen. So good afternoon, everyone. It is my great pleasure to talk to you about some of my research work in this meeting, together with all these excellent young scientists. And today the title of my talk is Modulating Vasculogenic Microenvironment for Tissue Regeneration. In regenerative medicine, tissue engineering holds a key position where three main factors come into play, namely stem cells, scaffolds, and signals or growth factors. In cell-based tissue engineering, stem cells play the most important role due to their capacity for both self-renewal and multilineage differentiation. And mesenchymal stem cells or adult stem cells hold an important potential for their possibility in use in clinical settings despite their limited differentiation capacity. And various types of mesenchymal stem cells have been identified in the orofacial region, out of which dental pulp stem cells from apical papilla, stem cells from exfoliated deciduous teeth are widely investigated for their regenerative potential. And these cells hold a promising potential in regenerative medicine for being readily available and can be harvested without causing any donor morbidity. So one of the critical challenges in stem cell-based tissue engineering is achieving an adequate blood supply for the survival of cellular or tissue graft until the regenerative outcome is achieved. And once we engineer tissue constructs, it is transplanted in vivo, and it solely depends on the oxygen supply via diffusion from the nearest capillary. And diffusion only serves up to 200 micrometers from a blood supply. The cells located beyond this distance are susceptible for ischemia and likely to undergo cell death. And ingrowth of host blood vessels is limited to a few tenths of a millimeter per day. Therefore, strategies to enhance vascularization of critical size engineered tissue constructs are required. And when we think of vascularization, two main processes are involved. Vasculogenesis is considered as the de novo creation of blood vessels from primitive mesodermal cells. These cells are converted into angioblasts, which form blood islands and then fuse to form the primitive vascular network. In contrast, angiogenesis is a formation of new vessels from the existing vasculature, and the initial steps involve degradation of the extracellular matrix, detachment of pericytes, and tip formation. So if we aim to modulate these processes during tissue regeneration, it is important to explore what cells are involved and what factors govern the outcome of the process. It has been shown that endothelial cells interact with mesenchymal stem cells, in angiogenic and vasculogenic processes, and therefore the microenvironmental factors of these cells play a key role. And cells are influenced by their interactions with the matrix, other types of cells, soluble factors, and physical or mechanical cues such as oxygen tension, compressive or shear forces. Therefore, engineering vasculogenic microenvironment that can recapitulate the favorable cellular matrix, soluble factor, and physical cue interactions is critical for successful outcomes in tissue regeneration. And accordingly, the methods used in enhancing vascularization focuses on incorporating factors that can mimic the natural vasculogenic microenvironment into engineered tissue constructs. So next, I will talk about these different microenvironmental factors and what our research findings revealed about those factors in relation to dental tissue regeneration. And many mesenchymal stem cell types, including dental pulp stem cells, are known to occupy a perivascular niche in vivo. At this niche, dental pulp stem cells interact with endothelial cells, pericytes, matrix, and also the soluble factors available. And in one of our early studies, we demonstrated that dental pulp stem cells and endothelial cells synergistically enhance odonto-osteogenic differentiation and angiogenic potential. And furthermore, we facilitated self-assembly of dental pulp stem cells and endothelial cells into 3D microtissue spheroids and observed that in the presence of dental stem cells, endothelial cells can arrange into vascular-like structures. And when these pre-vascularized dental pulp stem cell microtissues were injected into root canal space of tooth slices and implanted in immunocompromised mice, it gave rise to vascularized dental pulp-like tissue. Dental pulp stem cells facilitated enhancing vascularization, while endothelial cells promoted the matrix formation and odontogenic differentiation during dental pulp-like tissue regeneration in vivo. So this study also demonstrated that 3D self-assembly facilitate intensive cell-to-cell interactions, which in turn results in a panel of biological and regenerative properties. For the generation of complex tissues, monoculture spheroids of different cellular origin can be combined, or alternatively, multiple cell types can be incorporated in co-culture spheroids. If we are to use scaffolding materials to encapsulate stem cells and endothelial cells, the materials should be able to facilitate the natural cell matrix interactions. So we hypothesized that if engineered tissue constructs can be pre-vascularized with a preformed endothelial vascular network, these pre-vessels can anastomose with the host vasculature once transplanted in vivo. In order to achieve this, the matrix had to be optimized for both dental pulp stem cells and endothelial cells to survive and functionalize into vascular network formation. So once injected into root canals of tooth fragments and transplanted in immunocompromised mice, vascularized pulp-like tissue was regenerated. The perivascular or the prevascular network was able to anastomose with the host vessels, showing enhanced vascularization compared with the non-prevascularized group. Although cell-to-cell and cell matrix interactions are critical in a vasculogenic microenvironment, physical factors could influence these functions in a drastic manner. For example, following transplantation into the root canal, the engineered cellular constructs immediately face a hypoxic environment, making them vulnerable to lethal damage caused by depletion of oxygen and nutrients. When the oxygen tension drops, hypoxia-inducible factors, mainly HIF1 alpha, are activated in cells and induces a series of adaptive mechanisms for this hostile ischemic environment. However, the cell survival is very much dependent on how fast the tissue graft can receive a sufficient amount of vascularization. And we hypothesized if dental stem cells can be preconditioned to hypoxia ex vivo, they will be more resistant to in vivo hostile microenvironment. As HIF1 alpha expression is stabilized, the downstream signaling could induce VEGF expression and exert angiogenic effects. So we stabilized the expression of HIF1 alpha in dental stem cells by silencing PHD2, which is an inhibitor of HIF expression under normoxia. This in turn resulted in enhanced VEGF expression, the most critically important growth factor in angiogenesis. And the direct co-culture, the fibrin bead assay and Matrigel assay demonstrated that the endothelial cells in the presence of HIF1 alpha stabilized dental stem cells formed significantly enhanced vascular tube formation. And after transplantation into the subcutaneous space of mice, the Matrigel plugs were examined for the vascularization. And as you can see here, HIF1 alpha stabilized shed with or without endothelial cells showed the highest amount of vasculature. And these results confirmed that the HIF1 alpha stabilization in dental stem cells can enhance the angiogenesis up to a level that incorporation of endothelial cells is no longer needed. And the results of this study showed that HIF1 alpha stabilization can enhance vascularization via promoting both angiogenesis and vasculogenesis through induction of endothelial differentiation. And subsequently, we used HIF1 alpha stabilized dental stem cells in an in vivo pulp regeneration model. As you can see here, the HIF1 alpha stabilized cells gave rise to pulp-like tissue with significantly enhanced vasculature, as shown by increased number of vessels, as well as the diameter of the vessels. So collectively, the current study demonstrated that hypoxic preconditioning by HIF1 alpha stabilization is a very promising mode of priming dental stem cells to achieve enhanced post-implantation angiogenesis using a single population of cells. So growth factors or cytokines secreted by stem cells and endothelial cells are key factors regulating different steps in angiogenesis. So it has been shown that VEGF is the key angiogenic factor that promotes initiation of angiogenesis. And once the vessels are formed, pericytes need to be recruited to the nascent vessels in order for the vessels to get stable and mature. And often the engineered vessels are immature and leaky. Without stabilization by pericytes, these vessels will eventually regress, causing vascular failure. So dental stem cells have been proposed to have the capacity to act as pericytes. However, the signaling that regulate the recruitment of dental stem cells as pericytes is poorly understood. In one of our ongoing studies, we proposed that once the nascent vessels are formed, SemaPhoD act on endothelial cells to stimulate dental stem cells to act as pericytes. So in this study, we use a microfluidic device to recapitulate the complex 3D in vivo microenvironment. As you can see, endothelial cells were seeded in the center channel, and dental stem cells were seeded on side channels so that they can migrate into the center channel in response to the signaling they receive. So you can see that the dental stem cells express pericyte markers even at their naive state. However, when cultured in SEMA4D treated endothelial conditioned medium, the phenotype changes to a more mature state with increased NG2 and SM22 alpha expression. And also we found that SEMA4D increases the total vessel length as well as SM22 alpha positive dental stem cell supported vessels. And the quantification of these SM22 alpha positive supported vessels revealed a significantly increased pericyte coverage in the SEMA4D treated group. And in higher magnification, we observed co-localization of endothelial vascular tubes with SM22 positive dental stem cells, confirming their pericyte-like action. And to further confirm the role of SEMA4D in this function, we silenced the expression of plexin B1, which is the SEMA4D receptor on endothelial cells, and repeated the experiment. And the results showed a significant reduction in pericyte or SM22 alpha positive supported, positive dental stem cell supported vessels. And in order to identify the endothelial derived factors which are responsible for pericyte recruitment, we performed ELISA on SEMA4D treated HERAX and found a significant increase in PDGF-BB, but not in other growth factors. So when PDGF action was inhibited by adding PDGF inhibitor, the dental stem cell supported vessel number was significantly reduced. So this study showed that SEMA4D could promote recruitment of dental stem cells as pericytes acting on endothelial plexin B1. Furthermore, PDGF signaling plays an important downstream role to act on dental stem cells to modulate its function as pericytes. So as I mentioned, the strategies of engineering the vasculature could vary from simply co-culturing of endothelial cells with mesenchymal cells to fabricating scaffolds with channels that can be pre-vascularized to 3D bioprinting of vascular cells directly on patterned hydrogels. However, the outcome still depends on many different factors that act together in a given microenvironment. Therefore, we need to optimize strategies for more reliable and predictable outcome and need to use novel strategies to identify optimal targets and molecules. And in order to translate the findings to the clinic, randomized controlled trials are required. So that's the end of my talk, and I would like to thank all of my mentors and collaborators, and also to my hardworking team, and specifically to Yvonne Han and Lily Zhang, who performed most of the laboratory experiments that I mentioned in the latter half of the presentation. And last but not least, the funding I received from the University Grants Committee and Food and Health Bureau of Hong Kong. Thank you for your attention. Okay, so Dr. Nam. So the next speaker is Dr. Nam Hyun.
Yes. Can you hear me?
Yes, I can hear you.
Okay, so I can start.
Yeah, you can start sharing your screen.
Okay. Okay, you can see it.
Yes, I can see your PowerPoint.
Okay. Sorry. Good afternoon. My name is Nam Hun. I'm come from the University of Medicine and Pharmacy at Ho Chi Minh City. And this is the view of my university. I graduated PhD from Chulalongkorn University for the Faculty of Dentistry in oral biology. And then I finished my postdoc in Department of Immunology at the University of Tokyo. And I just come back from Tokyo to Vietnam recently. I finished some my research in the single cell mRNA sequencing analysis in human, focus on bone biologies and osteoimmunologies. So another study that we discovered the pathway during the osteoclastogenesis at a single cell resolution. And in these studies, I cooperate with Associate Professor Tay Huan from the Institute of Oral Medicines, National Cheng Kung University, Taiwan. And this study was funded by the UNP and NCKU Joint Research Program. The topic is about a single-cell whole transcriptome analysis in oral cancer. So we know that oral cancers in general is one of the most serious disease of the human. And more, the number of patients increase, especially in the Southeast Asia and Pacific Asia area. In dentistry, the left oral cavity cancers take very important and is increased every year, especially in Asia. We see that in the figures, both incidence and mortalities in Asia, they are number one in the case number. And molecular diagnosis of oral cancer and oral potential malignant disorders, we have many methods that we can diagnosis from the clinical, at the tissue, the cell level, into the DNA, RNA, protein level. However, the more accuracies, the more expensive. And some methods look like the next generation sequencing or the microarrays is quite expensive. And the technology is also very complicated. We know that in our whole body, we have around 37 trillion cells, and each cells have is located in its tissue, its organs, and they have various very diversities functions. Previously, we have a box mRNA sequencing that we pull on the cell in the tissue, and we have the average expression of the gene. However, with the development of technology, we have a single cell RNA sequencing that we can sequence the genome in the transcriptome in each cell. Recently, we have another study about a single-cell transcriptome analysis of the primary and metastatic tumor ecosystem in the next cancer. And they found that the single-cell RNA sequencing reveal a diverse malignant and stromal and immune cells of the tumor. And the malignant cell vary in cell cycle, hypoxia, and epithelial expression programs. And partial epithelial mesenchymal transition program, we call it PEMT, at a leading edge is regulated by the microenvironment of the tumor. and computational modeling refined of the cancer cell genome atlas subtypes, linking the PEMT to the metastasis. In our study, we hope to identify the new marker of the head and neck cancer, the oral cancer, and with the power of computational methods, we hope to have diagnosis and treatment as well as the drug prediction, and leading to the future of the accurate the target medicines. So we take the sample from the head and neck cancer, we do the clinical parameter, also the single cell RNA-seq, and then we connect with the immunohistochemistry so to identify the new biomarkers and drug prediction. However, this study is still going on, and I hope that I will have more data in the future to present to everyone. First, we start with the oral cancer mouse model. The mouse, the oral tumor cell of the mouse, we have 4 groups at 16 and 29 weeks, where we call the control, and the cancer-induced, we call the treatment group. So after the qualified control, we retain the 7,000, around 8,000 cells, and unbiased clustering, we results in the 21 cluster. So, and the cluster was identified by canonical markers. So you can see in the figure on the right, we have an immune cell look like a T cell in the orange color. I will show the okay, and we also have granulocyte macrophage as well as the fibroblast, keratinocyte, endothelial myoblast. So sorry. Well, okay, sorry. And the average expressions of the canonical markers that were shown on the figures, we can see that's for the example T cell. We have the expression of CD45, CD3. B cell, we have expression of CD19, resident macrophage 1, 2, circulation macrophage, as well as granulocyte, different C18 granulocyte, neutrophil, endothelial, fibroblast, myocyte, myoblast, anthrocyte, epithelial, and keratinocytes. So, as I explained, that's with the traditional method, that bulk RNA sequencing, we have the average expression of all the genes in the tissue. So with single cell RNA sequencing, we have the expression of the gene in at single cell level. So I can explain how it's different in here. For example, from the single cell RNA-seq data, we can calculate the pseudo bulk RNA-seq result. It means the average. We calculate the average expression of all the cells, and we have expression average expression of the cell in the control. It means the healthy group, and also in the cancer induced group. And we just have 755 genes. That's a significant difference. And some gene in the figure that's in the blue color, collagen HIF1A is just example. And when we do the enrichment analysis, we can see that's how the difference of the pseudo bulk RNA-seq show in the cancer-induced. On the left-hand side, we see that's cancer-induced, or the treatment group. We have increase of the malignant neoplasm of the mouse, the inflammation, and we check for some signature gene On the right-hand side, we see that some cancer genes look like ANSER1, SPLIP, SLP, MMP9, for example. But that's all. The information is very limited. So when we look at the real single-cell RNA result, we can see that in each cluster, each cell type, we have different signature genes as well as different the enrichment analysis. For example, on the left-hand side, we can analyze that in the keratinocyte of the treatment group, we have increase of some transcription factor, especially the MYC. Is the master transcription factor of the cancer. And on the right-hand side, when we analyze the disease associated with the keratinocyte in the treatment group, we see that it's increased in the carcinoma, carcinoma spindle cell, carcinomastosis, leukoplakia, etc. So we can see that's an advantage of the single cell analysis. And for another example is the epithelial 1 and epithelial 2. We can see that the transcription factor that control the cell, the cells in the treatment group. That we have many transcription factor. These own transcription factor is very famous in the control the cell cycle, control the cell death and cancer. And with the disease analysis, we also see that epithelial 1 and epithelial 2 enriched in some squamous cell carcinoma, mammary carcinoma, undifferentiated carcinomas. So with the single cell analysis, we can focus on which cell type is associated with the cancer group, and we can discover the transcription factor that control the gene expression in that group, and with the type of disease that we can focus. And then we also investigated the gene that's increased in the cancer group compared to the treatment group. So we have that, this is the example genes. We have as many genes that associate in the control group, and it's different in each cluster. It means it's different in each cell type. For example, CYTID4 is the genes that control the cell cycle. PCAM, DNBT1, or SRA1 is very important gene in cancer. But the problem is that we want to find the new gene. That's the gene that have a less function, less well-known function that's associated with the cancer. That we want to discover the new biomarker. So let's see what we discover in human cancer. In human cancer, we intend to do many groups. Let's see, candidates, OP, MD. This is the pre-cancer group or candidate with the squamous carcinoma cell, or non-candidate or healthy sample. However, this study is still going on, and we still recruiting the patient. And now we have first we have to finish data is the OP, MD, and SCC with the candida infection. And we also, similarly to the mouse, we also identify the many cell type in the human sample. And we can identify clearly a lot of immune cell in the tumor of the human. If we have a CD4 T cell, CD8 T cell, NK cell, B cell, plasma cell, macrophage, and we also have myeloid cell, MAC cell, endothelial, fibroblast, epithelial, keratinocyte, monocyte, translating cell, and the cancer cell. And similarly to the mouse, the animal, we also identify many genes that's associated to associated to the human to the cancer group. And we focus in cells a gene that's called LAMC2. The LAMC2 is a gene that's associated with the ECM, the extracellular matrix of the cell. And in mouse, we cannot find the significant increase of this LAMC2 in the mouse model. However, in our data, our ongoing data, we can see that LAMC2 increase in the SCC, the squamous carcinoma cells, and analyze previous published data. We also found that LAMC2 increase significant in the malignant cell. And when we do some immunohistochemistry, on the left-hand side, we can see that in the normal, the healthy patient, we see that LAMC2 just on the basal membranes, not inside the cell, but in the oral cancer carcinoma, squamous carcinoma cells, we can see that LAMC2 expressed everywhere, even inside the cell. And with the lymph node metastasis and non-lymph node metastasis, we can see that different pattern expression of the LAMC2 with different type of invasion, type 1, 2, 3, 4, and this have a very different expression pattern. So this study is still going on, and when we finish the whole data, we can show that the importance of LAMC2 during the oral cancer development. Because the time is not so much, so I feel thank you for your attention, and I hope that when I have more data, so nextly the next plans up after this one, I and Dr. Professor Huang try to collect all the patient sample that we intend to do, and we will analyze in a large number of the patient about the expression of LAMC2 to see how it's different during each stage of the cancer with this marker. And we hope that we can contribute to the knowledge about the oral cancer research. Thank you so much for your attention.
Thank you, Dr. Nam, for your very informative presentation. And our next speaker is Dr. Citra Theodoria.
Good afternoon, Dr. Waruna.
Yes, hi, Dr. Citra.
Hi.
So
nice
you can share your screen. Yeah, nice to see you too.
Okay, so now can you see my screen?
Yes, we can.
Okay. So may I start now? Okay.
Yes. Okay. Thank you.
Thank you. Okay. Hello, everyone. My dear professors and colleagues. It is my privilege to be here. And first of all, I would like to thank to the committee for this opportunity for me to be able to join in this Young Scientist Forum at IDR Southeast Asia Division Scientific Meeting 2021. It is an honor that I can see many outstanding researchers, and I think we enjoy. And my name is Citra Fragancia Theodoria. I'm from Department of Oral Biology, Faculty of Dentistry, Universitas Indonesia. And in this time, I would like to share my presentation entitled A Game Changer of Oral Microbiome Related to Non-Communicable Disease in Indonesia. So I would like to dedicate my presentation to my dearest teachers, seniors, and friends that cross the same path with my journey. And in my presentation today, I will divide it into two main topics. So first, I would like to share a glimpse of non-communicable disease and global health and the challenges opportunities of oral microbiome studies in Indonesia. As you know that non-communicable disease, known as a chronic disease, as one of the major challenges for health and sustainable human development. And if disease frequently referred to communicable or non-communicable. Communicable disease comprise infectious disease such as tuberculosis and measles, while non-communicable disease are mostly chronic disease. And to date, the global NCD's response has focused on 4 major NCDs. They are cardiovascular, heart disease, cancer, diabetes, and chronic respiratory disease. However, there is a range of other conditions such as congenital neurological disease, obesity, stunting, dementia, mental disease, wasting, metabolic disease, include the oral disease that link to the 4 most prominent non-communicable disease. Non-communicable disease disproportionately affect globally. Furthermore, non-communicable disease are characterized by a long disease phase, and then it became leading cause of death in our society. And Indonesia has high prevalence of non-communicable disease. Indonesia has a 4th population in the world, the biggest 4th population in the world, which consists over than 200 million inhabitants, including numerous ethnic, culture, and linguistic groups. And nowadays, Indonesia is struggling to deal with the non-communicable disease. Since 2010, the leading causes of mortality and morbidity in Indonesia have been dominated by NCDs, such as cerebral stroke, heart disease, diabetes mellitus, and impaired development. And we are known that Indonesia, known as a representative of Southeast Asia in terms of lifestyle and diet, notably a rice-based daily diet. And Indonesia is the home of diverse cuisines and traditional fermented food, and blessed with mega biodiversity, preserving perishable agricultural products by spontaneous fermentation, such as tempe and dadi, that has been carried out by since the 17th century, involving mixed cultures, including lactic acid bacteria. And the fermented foods become an integral part of the diet prepared in household or the home industry, using relatively simple technique and equipment, and contributing not only to preserve food, but also providing bioactive compounds, nourishing the gut microbiome, and in turn, improving the nutritional status, while at the same time bolstering economic development. And by 2030, about 70% of Indonesia's population will be of working age. So it will be support the windows of opportunity. Healthy, safe, and nutritious food should be available and accessible to all. So it provides the healthy and productive community and reducing healthcare expenditure by suppressing the both acute and chronic malnutrition, infectious disease, and non-communicable disease that will allow individuals to live happier and more prosperous. So this food development will be challenging by valuing the local food and plant biodiversity and so on to the characterization or profile of the microbiome. When you see this title, it is well known that microbiota dysbiosis is closely associated with numerous diseases in the human body. The oral cavity and gut are the 2 largest microbial habitats, playing a major role in microbiome-associated disease. Even though the oral cavity and gut are continuous regions connected through the gastrointestinal tract, the oral and gut microbiome profiles are well segregated due to the oral-gut barrier. However, the oral microbiota can translocate to the intestinal mucus in conditions of the oral gut barrier dysfunction. Inversely, the gut to oral microbial transmissions occurs as well in inter and intrapersonal manners. And recently, it has been reported that oral and gut microbiomes interdependently regulate physiological functions and pathological processes. Oral to gut and gut to oral microbial transmissions can shape or reshape the microbiome ecosystem in both habitats, and eventually modulating the pathogenesis of the disease. So when I tried to find some study report regarding the microbiome study in Indonesia, it was very limited. However, one thing that's interesting here is this study cover microbiome in specific region of Indonesia province. You can see here there are Yogyakarta and Bali, Makassar, Yogyakarta also, and Banten and West Java, and also West Sumatra. On the right figure, on the right information studies that you can see here, West Sumatra mostly provided with the meat and curry and coconut milk based, while the West Java carried by plain salad and rice. But here you can see there are differences based on the geographical units, the location. So as you know that the oral microbiota can be classified into a core microbiota and a variable microbiota. The core microbiota is the same for all individuals, while the variable microbiota is different between individuals in response to unique lifestyles and phenotypic and genotypic determinants. The major genera with the largest representations in oral cavity include Streptococcus, Haemophilus, Rosia, Prevotella, Porphyromonas, Veillonella, Neisseria, and Actinomyces. The oral cavity contains over 700 microbial species, as well as commensal and opportunistic microbiome, which organize into different microbial habitats. Therefore, the oral cavity is a very complex habitat because there is a unique and diverse microorganism community, regardless of the niche locations of all oral sites of the healthy subject can carry core oral microbiome. In addition to this common bacterial niche, each niche has a well differentiated between the bacterial composition, such as harpalate, buccal, tongue, supragingival, subgingival, and labial, saliva, and gingival clavicular fluid. So the dental hygiene, I think, is very important factors that may shape the oral microbiome, since the oral cavity it will directly open to the outside of the environment. Indonesia is the fourth populated country in the world and has 7 main islands, such as Sumatra, Java, Kalimantan, Lesser Sunda Islands, Sulawesi, Moluccas, and Western New Guinea. And these main islands contain 34 provinces with many heterogeneous factors that I mentioned before. However, oral microbiome study in Indonesia is very limited. So to be honest, so far that I searched in PubMed, there are 2 original research regarding the oral microbiome study in Indonesia. So here you can see the diversity of oral microbiome of women from urban and rural areas of Indonesia, and also in this case on the right, the population in Jakarta. So when I tried to search the information, it was very limited, and this figure that I share, I want to tell that this review provides a comprehensive account to such geography, ethnicity, and lifestyle-specific variations in healthy microbiome at the 5 major body habitats. Here you can see there are gut, oral cavity, respiratory tract, skin, and urogenital tract. And this review focus on the general trend in the human microbiome evolution and a gradual transition in the gross composition structure along with the continual decrease in diversity of the microbiome. So I tried to sum up some papers, and this paper of oral microbiome, which has related to non-communicable disease. So from here, you can see in Indonesia, the healthy women, the oral niche that they isolate from tongue, and you can see here the diversity. Those informations provide that the oral microbiota of the urban localities significantly richer in the bacteria than rural localities. And our study regarding the normoglycemic prediabetes and type 2 diabetes, we found that the type 2 diabetes patients has more diverse than pre-diabetes and normoglycemic group when we isolated from saliva. And it's also supported also by this study from Chen. So they also compare the healthy and type 2 diabetes from buccal mucus, and they mentioned that they report that the type 2 diabetes samples has significantly higher in the diversity. And in contrary, from Saab et al., they isolated from saliva regarding the normoglycemic prediabetes and type 2 diabetes. They report the reductions of biological and phylogenetic diversity at the diabetes and pre-diabetes compared with the normoglycemic status. And Catherine et al. Also mentioned that the type 2 diabetes is more diverse than healthy. So this fact that the susceptibility to this disease often shows that the ethnic or the location or the geographic that inspired the investigator to explore the geographical variations in oral microbiome and its potential impact to the oral health. So actually this collectively results that revolutionized the field of oral microbiology because they have revealed a degree of biodiversity among oral bacteria which has exceeded expectations. The dynamic of oral microbiota are now better understood because of the identification of patterns and transmissions of bacteria. But one of the fundamental issues in the microbiome research is the characterization of healthy human microbiota, which diverse based on the geographic variations, different race, different ethnicity, and lifestyle, urban and rural, or maybe habit of smoking or alcohol and so on. And also this molecular approach that the uncultivated microorganisms still not revealed. I mean, it's difficult to cultivate, so it's difficult to reveal the composition of the profile of the microbiome. And finally, I think that some of geographical or racial variations in microbiome structure have been attributed to differences in host genetics and immunity, while in many other cases, culture and behavior like diet, hygiene, and so on, it can overshadow the genetics. So it could be our opportunities that we can assume that the microbiome-based therapeutic strategies will be studied in the future, and it will be recommend the geographic tailored community scale approaches for the microbiome study. So here's the conclusions of my review, that oral microbiome in both localities, many localities have a unique tendency of species to co-occur and interconnections between its member to sustain the ecological balance. And the prospect of functional food development, it will be very challenging by valuing local food, plant biodiversity such as roots, fruits, vegetables, and so on. And I think that the important thing that we can found the innovations in food technology and nutrients, it will be required, which will ultimately help deliver a range of functional food applications that meet nutritional needs for the consumers. So, and the organisms of ever-evolved culture-based and state-of-the-art in the culture independent of the molecular technique, it could be facilitated in-depth understanding of the dynamic acquisitions and transfer of oral bacteria. So I think that's all my review regarding the oral microbiome in Indonesia related to the non-communicable disease. Thank you very much for your kind attention.
Okay. Thank you, Dr. Citra, for your very nice talk. And so we have a few minutes for questions. So I would like to request the audience, if you have any question, you can type in the ask a question box on the right side of your platform screen. And so until we receive some questions, I would like to start. Maybe I can ask a question from Dr. Sriram. Dr. Sriram, can you hear me?
Yeah, yes, okay.
Yeah, so I noticed that you have done some work with pericytes, then some fibroblasts and endothelial cells. So may I know what is the source of pericytes you used?
All right, so yeah, we tried with different sources. Like for the pericyte work, we used was using embryonic stem cells and pluripotent stem cells. Whereas for the fibroblast, it's something like very much depending on whether we are working with skin tissues or gingival tissues or say oral mucosal, then we have those specific fibroblasts, the region specific fibroblasts.
Okay. Yeah. So, and also I saw your video showing how the pericytes go to the middle of the spheroid and endothelial cells at the periphery of the spheroids. So
yeah.
Do you know exactly why it happens, or like when it will change to the other way? I mean, when it will, because usually in blood vessels, we see pericytes outside of the endothelial tubes.
Yeah, excellent question. That's something like is sort of dependent on the size of the spheroid itself. Like if you see like these spheroids, and the size of the spheroid again is dependent on total number of cells what we have. And so that is one of the factor, like the number of cells and the size of the spheroid. And then another aspect is the ratio between endothelial cells and pericytes, or even say fibroblasts for that matter. And even MSCs. So they all have very similar characteristics. So the ratio between endothelial cells and these mural cells, they also determine it. When you have a low number of cells, total number of cells, the spheroids are smaller. And in that case, the endothelial cells like to go to the periphery. But if you have a much bigger size and you have sizable amount of the mural cells, then these mural cells would secrete matrix as part of their 3D culture model. And also there's going to be a certain amount of hypoxia. And this eventually attracts the endothelial cells to go into the core of the spheroid. And then you can end up having the mural cells on the outside and the endothelial cells in the inside of the spheroid. And here, like, they are just not going to be sitting just one end to end, but instead the endothelial cells like to form networks, vascular networks within the matrix, which is laid down by the mural cells. So it's sort of dependent on these other parameters as well.
Yeah. Okay, so those spheroids. So what is the maximum size or the average size of a spheroid you formed?
All right, so the ones what we work is around like 200 to 300 microns. But like we have worked almost up to close to a centimeter, close to a millimeter. But of course, like in a millimeter one, like we have these necrotic centers, right? And so that's where like we are now working with the microfluidic systems so that like we can have a more active perfusion, and we see that then we have the ability to form much bigger spheroids.
Yeah, thank you. Very exciting work.
Yeah, thanks.
So,
all right, I had some questions for you again, like for the work with the microfluidic platform, right? Like with the endothelial cells and DPSCs, and as well as like the pericytes and so on, right? So in that work, like because these are microchannels, like what kind of matrix do you use?
So in the center channel, we use fibrin gel. So fibrin gel supports the vascular tube formation by endothelial cells. But on the side channels, we don't use any matrix. So it is just a single layer of cells. But,
yeah, so that's excellent. Like that fibrin is so good, right? But,
yes.
Do these endothelial cells form like luminized structures which connect from one lumen to the other lumen?
Yes, it does. So basically, depending on the time point, the endothelial cells can form vascular tubes connecting with each other from one side channel to the other side channel. I mean, so it kind of extends in the whole of center channel.
Yeah, excellent. So that means it also brings opportunity to study on immune cell migration, right? And maybe like even metastasis, working with cancer. I think like NAM is working with cancer, right? Like so you can like work to have cancer metastasis models.
Yeah, exactly. The microfluidic devices kind of facilitate many in-depth studies. I think we are almost reaching the end of our session. So thank you very much for all the speakers. Your contribution was very important to the success of this forum. Okay, so if no questions, so there will be the annual general meeting of the SCA IADR division in the same room. So that will be oral session hall 1. So I invite all the speakers and all the audience to attend the meeting.
Thank you, Dr. Warna.
Yes, sure. See you all.
And thank you all.
Prof. May, this is the tech team. We will take a 5 minutes break before the next streaming.
Thank you very much. So for the other speakers, I think if you don't mind, we thank you so much for your talk, and then we need to get this room ready for the AGM. So would you please leave the room? Thank you.
Prof May, you will have to use a different link to join the AGM. That's the keynote.
Really?